Uh, Good morning, everyone. My name is Rakshika. I am a PhD student here at SGPC, and I will be your host for the day. I hope you all had a really good dinner last night and hope you had good rest. And we will now be starting day 3 of the third BRICS Neuroscience Symposium. I would like to start off the day by reiterating the housekeeping rules. There'll be 14 sessions across 3 days, and each of them would, today we are going to have 3 scientific sessions. Each of them are going to have like 2 to 3 talks, followed by 10 minutes of question and answer. There will be one panel discussion today, followed by closing remarks, and we will be heading for the Shore Temple visit around 3:15 p.m., where we will assemble downstairs for the buses. So I would like to start off the first scientific session for the day, where we are going to have Professor Vidita Vaidya, Dr. Pratik Chaudhary, and Dr. Joshua Davimis. So.
Now, you can hear me now? What? Sorry, now you can hear me. I can hear the echo back, yes. Okay, good morning, everyone. I have the job of being the opening batsman, so to speak. But what I'm going to do in the next 15 minutes is give you a little bit of a glimpse about why there's such a lot of buzz around this word serotonergic psychedelics, and why people are so excited about these molecules that have actually existed for thousands and thousands of years and have also been used for thousands of years. So if you look at the ethnobotanical usage of psychedelic-like molecules which produce hallucinations, I think the thing that becomes most obvious is that amongst all the different plant and fungi-derived molecules, there's this one tall pillar, which is hallucinogens from Native American civilizations. And so they've been indigenously used in the Amazonian tribes. They've been used in diverse cultures across the world, North Africa, Southern Europe, India, Brazil. We heard a little bit about that from Thai yesterday. So there is an indigenous usage of these molecules for a long time. But in a sense, nothing really happened in the modern Western version of looking at these molecules until they were synthesized or extracted, first with mescaline in the late 1800s and early 1900s, and then the big sort of event was actually the synthesis of lysergic acid diethylamide, which happens in 1938, and in 1943, Albert Hofmann tries it on himself, which is when we have the first sense that we've actually synthesized a molecule like LSD, which produces very potent hallucinations. And then through a period of time, there is, of course, a therapeutic exploration of these molecules. But about the 1960s, you have both clinical research and recreational use simultaneously hitting each other. And then these molecules end up in 1970 banned as Schedule I molecules, Substance Abuse Act in the US. And they're all still Schedule I molecules, most of them, which means that they are banned and they're illegal. But there has been, in the last 10 years, a resurrection or a revival of interest in these molecules, partly because mental health conditions are a serious burgeoning crisis for the world, and the quality of treatment currently available has serious handicaps. Most antidepressants, antipsychotics, and other drugs are slow to work, slow to act, and there's quite a bit of treatment refractoriness, which only underscores the need for better quality drugs. Of course, these drugs come with, it's a mixed bag in that you sometimes get very important and relevant therapeutic-like effects, but you also produce psychotomimetic actions with these molecules. So what I'm going to do is tell you a little bit about what's currently going on in the field in serotonergic psychedelics, and also tell you a bit about what I do in my laboratory. So if you just were to do a search for the term serotonergic psychedelics and look on it on PubMed, this is what you would see, that there was this initial burst of interest, then there's a complete shutdown for a very long time after 1970 because of the banning of these molecules, and then something has happened, and this only keeps climbing upwards. Something has clearly happened in the last 10 years that has resulted in a significant revival of interest in these molecules. Just to give you a sense as to what these molecules are, this is what they look like. Okay, so the one that you would know is the naturally occurring non-psychedelic neurotransmitter that many of these molecules mimic. That's serotonin. Psilocybin is an ingredient of magic mushrooms. Dimethyltryptamine, you heard about from Thai yesterday, which has been used. It's the active ingredient of ayahuasca and many other plants. Mescaline, the active ingredient of the peyote cactus. LSD, which is synthetic. And you can see that the orange ones are the ones that produce hallucinations. And then there are similar structures which do not produce hallucinations and are ligands at the serotonin 2A receptor. The molecules we work with in the lab are DOI and Ariadne. It's a phenylethylamine, again, a synthetic psychedelic. And you can see that they're very structurally similar, but this molecule produces potent hallucinations, and this one actually doesn't produce hallucinations at all. And you know, it's an interesting mix of people who are interested in these molecules. There are those who are interested from a psychotherapy clinical perspective. There's a substantial recreational usage which is interested in the narratives of sort of the psychedelic trip and the experience. And then there's a bunch of molecular, cellular neuropsychopharmacologists who want to understand exactly what is going on in the brain when you look at these drugs. And so it's the community of biomedical researchers who are interested go from wanting to understand the structural effects of these drugs at the molecular level on receptors, all the way to looking at cells and circuits, and all the way to networks. Again, you heard yesterday very nice work from Thai talking about EEG and how this gets regulated by DMT. The biggest challenge in our field, though, is that it's very difficult to jump across these levels of organization. So you may get a lot of mechanistic detail at one end, but you find it very hard to integrate this information across levels of biological levels of organization. Okay, so what are we doing in my lab? And I'm just going to give you a glimpse of a couple of the things that we do. To start with, we don't work with human material, very limited amount of work with human material that we do with Biju, Vishwanath, and team at NIMHANS, but most of what we do with rats and mice. And they respond to psychedelics as well, and we are interested in the hallucinatory effects of these drugs, their effects on anxiety, despair, fear. We're interested in their cellular effects at an organelle level, and we're also interested in what they do to plasticity. And I'm going to tell you about glimpses of two stories where we find that psychedelics appear to be very potent boosters of mitochondria in cortical neurons, and I'm also going to tell you about some of the circuit level understanding we have about anxiety. So this is the summary of the story. If you don't want to see the data and you walk away with the summary, it'll tell you the primary take-home message. Sashena, a very talented former postdoctoral fellow and now a research associate professor, assistant professor associated with my lab, made this very interesting serendipitous discovery that serotonin and serotonergic psychedelics, three of them here, DOI, TCB2, and NBOH, seem to, through the 5-HT2A receptor in cortical neurons, through a pathway that now we have well characterized, drive up the production of mitochondria in cortical neurons. And here's the data. So if you treat rats with DOI, the psychedelic, and look 2 hours, 6 hours, 24 hours later, you will see that the mitochondrial DNA content, which is one way of reading out mitochondrial mass, goes up in animals within the cortex. And it appears to be particularly unique to the cortex because we don't see these effects in the hypothalamus or the striatum. We do see them in the cerebellum, which is super interesting. So the two circuits that are showing a strong response to psychedelics in terms of mitochondria going up are the cortex and the cerebellum. And you see associated with this a slow but emerging rise in ATP levels in the brain as well. And then if you go in and isolate mitochondria now from the cortex of these, of these animals that have seen DOI, and you measure oxygen consumption rate or look at basically cellular respiration of mitochondria within these animals, what you can clearly see is that if you've been treated with a psychedelic, you will see this boost in state 2 respiration, state 3 respiration, and ATP production rate. Okay. Moving from the data, let's start asking, what does this actually mean, right? So what we found, and in a sense we stumbled upon, is that psychedelics, and I showed you one example, but we've looked at multiple, have a fast, quick effect on mitochondria, driving up their production and enhancing energy within neurons. Now, the question is, what does this even mean? What does it mean to increase the availability of, enhanced bioenergetic capability in a sense. You'd also worry about whether that produces ROS, but it turns out it actually is ROS scavenging. And these animals, these neurons are actually able to buffer stress significantly better. This is a serotonin 2A receptor mediated effect. You knock out the serotonin 2A receptor, you lose all these effects. So it's 2A receptor mediated. And here's the pathway. I'm not going to show you all the data, but I'll just show you the results so that you can see what we find. What we found is we narrowed down the pathway to show that it's 2A receptor dependent. You knock out the 2A receptor, you don't get the effects. It's mediated through a cascade that involves GQ, PIP2 hydrolysis, calcium, and then growth factor release. So BDNF gets dumped out from these neurons after they see the psychedelic. And that BDNF, through a cascade, won't go into the details, but we know the steps, drives up more mitochondrial production. If you do not allow BDNF to work by blocking the TrkB receptor, you lose all these mitochondrial effects of psychedelics. That's in vitro, this is in vivo. You can actually go into the prefrontal cortex, prevent the action on TrkB receptors, and you will lose these mitochondrial effects. So what we think is going on is serotonergic psychedelics drive up trophic factors, quick, fast effect. That seems to drive up mitochondrial production and OCR. And this is a potently neuroprotective action because these neurons now buffer oxidative stress and excitotoxic stress significantly better. Not showing you all the data, but we really think that this is one of the big neuroprotective actions of these molecules. So I'll switch then to telling you the other story. So that gives you a sense of what we are studying at a very cellular scale, right? Where we can do this in neurons in culture as well as in animals in vivo. And we're exploring precisely this with Biju in neurons now in humans. So iPSC converted human neurons. And this is something that would be interesting to see how much of this holds true across different species. So now switching to a completely different scale, which is to ask what happens to mood-related molecules. This is the one big holy grail question, which is, can we use rodent models to study the comparative behavioral effects that happen in humans? Some of the behavioral effects reported in humans have a counterpart in rodents. Some of them don't have a good counterpart in rodents. So this becomes, it's a bit of a mixed game. And the question we were interested in asking is, it's been known for a while that psychedelics can have a therapeutic effect on anxiety, but we don't know which part of the brain produces this particular behavioral effect. Can we actually start pinpointing the circuit and the kinds of cells that mediate these actions? So this is work done by a very talented former graduate student in my lab, Prachi Tiwari, ably assisted by a current graduate student, Mahati. And Prachi is now at Johns Hopkins and hoping to make her way back to India to a faculty position. So just to give you a sense, what Prachi set out to do was to ask, okay, if we see an effect on anxiety, can we first pinpoint which part of the brain produces this behavior? And then if we can pinpoint that, can we go down and figure out which are the neuronal cell types in the microcircuit that actually drive this particular behavior? The reason this is so important is that the hot standing debate in serotonergic psychedelic field is, do you need the trip to get the beneficial mood-related effects? Or can you potentially separate the trip from the mood modulatory actions? Are these something that you can dissect out? Not that that debate has been resolved, but it's important to understand which part of the brain produces which effects. So this is what Prachi set out to ask, and here's what she showed. And I'll just summarize the data and then show you the little bit of a glimpse of the actual results. So what she set out to ask is, if you take rats and you systemically administer them a psychedelic, will it reduce anxiety-like behavior? And the way we test anxiety-like behavior in this particular example is elevated plus maze, but there's a battery of tasks that you can do. The elevated plus maze is a maze which is raised 2 feet off the ground. There are 2 open arms and 2 closed arms, and obviously self-respecting rodents would much rather spend time in these dark corners, hidden away, rather than in open, high elevated, bright areas where they may potentially fall off. So when you give rats or mice psychedelics, they seem to override any inherent fear of this space and actually end up exploring the open arms significantly more. What Prachi was able to do was to pinpoint, I'll finish in a minute, was able to pinpoint that this is driven through the ventral hippocampus, through a class of neurons called paravalbumin inhibitory interneurons, and it's these neurons that then seem to drive this particular behavior. And this is just to give you a sense of that. Here in rats, and in mice, you give them DOI, and you get a clear increase in the time that they spend in the open arms, doesn't matter if it's male or female, rat or mouse. And what she was able to show by actually very painstakingly delivering the drug to different parts of the brain, it is not produced in any of these brain regions. In fact, it's produced in the ventral hippocampus. So in the ventral hippocampus, in both rats and in mice, when you deliver the drug directly into the ventral hippocampus CA1 subiculum region, it is enough to produce this behavior. And if you block the serotonin receptor in this part of the brain, you lose these effects. And I'll just quickly tell you that the class of neurons, this is wonderful work which we collaborated with Alex Kwan and Antonio Ruiz. So I'll quickly tell you that bottom line, we figured out which class of cells is responding. If you drive this class of cells, it's sufficient to produce the behavior. If you inhibit these neurons, it's enough to go in the exact opposite direction. And the bottom line is that what serotonergic psychedelics appear to do is that when they go into the body, they go everywhere, obviously, but they also go to the ventral hippocampus, where they activate inhibitory interneurons, inhibiting the ventral hippocampus. And inhibiting the ventral hippocampus is enough to produce an anxiolytic action. Here, for example, there's no psychedelic on board, but we're simply inhibiting the projection neurons from the ventral hippocampus, and that is enough to reproduce the sort of effect that psychedelics give. All right, so this is the bottom line, in a sense. As I said, one of the challenges is to make it across different levels of organization. I showed you work that serotonergic psychedelics appear to increase their anxiolytic actions through these paravalvular neurons. Can you separate the hallucinatory effects from the anxiolytic actions? We don't think the hallucinatory effects are going through the ventral hippocampus, but we yet don't understand the circuits that produce all the hallucinatory-like states. Which circuits drive hallucinatory actions, antidepressant effects, and modulation of fear-related memories, and these mitochondrial effects that help enhance plasticity and survival. Is there a relationship between these very fast cellular effects and the eventual effects on mood? So wrapping up, that's the lab, the Hartman lab at the southernmost tip of Mumbai, most wonderful city in this country. Hopefully some of you will have a chance to visit, and this is the wonderful team that I'm very privileged to work with. I am done. Yeah.
Thank you, Dr. Vaidya. Now I call Dr. Pradeep Chaudhary to give his talk.
Good morning, everyone. I'm waiting for my slides. So, in recently concluded BRICS Health Working Committee meeting, mental health was recognized as one of the priority areas. So, I thank Richa and our colleagues at HGBC to including talks on mental health during the symposium. So, I work on early life adversity and how it impacts the aging trajectory and how can we reverse these trajectories. So this work was carried out in Vidita's lab at TIFR Mumbai, which led me to join HGBC a few months back. So the work was funded by TIFR, India Alliance Early Career Fellowship, and Sri PVF Foundation. So early life environment is very crucial in the development of neurocircuits responsible for emotional and cognition. Any adverse events during this early life may impact physiological as psychological functioning in negative manner, which can include social impairment, cognitive decline. In some cases, it can result in early death as well. So the bottom line is, more the number of adverse childhood experiences, more the chances of having negative health outcomes. So if you look through biological lens, so early life stress or early childhood stress may impact several biological processes, including stress response pathway, immune regulatory pathway. It can impact epigenetic changes, neurodevelopmental disruption, which lead to increased mood-related disorder, increased anxiety, neuropsychiatric disorders, and cognitive disabilities. So this is true across the species. It has been seen in monkeys, rodents, as well as in flies. So impact of early adversity has a long-lasting imprints in various animal models as well. So most of the groups study impact of early stress with respect to neuropsychiatry. They look at mood, anxiety-related behaviors, but very few groups focus on its impact on aging. So that is a key hallmark of early life stress, which occurs later in life. So you can see the history of early adversity changes the trajectory of aging as compared to the healthy aging. So, but the molecular and cellular players involved in this cascade are not clearly known. So we looked at the classical hallmarks of aging, and we pointed out, let's look at mitochondria and inflammation with this regard. What is the contribution of inflammatory pathways and mitochondria in early life stress evoke accelerated aging? So we focused on basically on hippocampal region of brain, and in the periphery we looked at the serum biomarkers. So how do we do early life adversity in rodent model? So we have used Sprague-Dawley rats, and there is a model called maternal separation, which includes separation of newly born pups away from the mother for initial 2 weeks of their life for 3 hours daily. So that has been shown to induce mood-related disorders, accelerated aging later in life. So here we confirm, first of all, whether the history of early life stress. When we do that, we allow these pups to grow older till their adulthood, middle age. And in this case, in middle age, we have looked at the cognitive behavior, which is a novel object recognition task. And we could see there is decrease in object recognition memory. So there is an impairment of object recognition memory in the MS group. MS is maternal separation, C is control. So control animals are not separated from their mother. So we see whether it is short-term or long-term memory test. There is a decrease in discrimination index, which indicates impairment of object recognition memory in the maternally separated cohort, which is middle age. The lighter dots here, if you can see, those are males, and the darker dots are females. So in males as well as females, we see the similar effects. Now, if we look at the same cohort and analyze their serum from these animals, then for the investigation of aging and inflammation-associated biomarkers, we could see there is increase in GDF-15. Now, GDF-15 is growth-derived factor 15, which is stress-responsive cytokine, you can say. So there is increase in aging-associated GDF-15. Again, C-reactive protein is the oxidative stress marker. There also we could see increase. I'm not going through all the results, but these are the examples. We have looked at several other markers of pro-inflammatory cytokines, where we note increase in those cytokines and decrease in anti-inflammatory cytokines. Again, we have looked at various markers of oxidative stress, metabolic regulators. There also we could see in maternally separated animals, which are in their middle age, show prominent inflammatory phenotype. Now, we have also looked at earlier time point where we investigated at mature adulthood, the animals which are 6 to 7 months old. So here we could see specific aspects of these systemic signatures are already apparent in mature adulthood. But not all signatures are getting upregulated or downregulated at this point. The severity is very reduced. So which cumulatively worsens as these animals with a history of early trauma ages. So this gave us a chance to do a cross-correlation analysis where we noted that the higher the cognitive decline, the higher it correlates with the higher inflammatory load in the maternally separated cohort, whereas in control animals we don't see any correlation. Given the inflammatory phenotype, we focused on neurovasculature, whether early stress impacts the blood-brain barrier integrity in middle-aged and adult animals. So we looked at using molecular, radio imaging, and biochemical techniques. So we could see there are different gene expression markers for the blood-brain barrier associated phenotype. So we could see only in middle age, there was a decrease in blood-brain barrier associated genes. In the biochemical technique, we injected Evans blue. Now, Evans blue generally does not cross the blood-brain barrier. If the barrier is leaky, it will cross, and we can detect the extravasation of the dye. So we use this technique, and we could see only in middle-aged MS maternally separated animals, there is increased extravasation of dye, indicating that there might be leaky blood-brain barrier. But if you see the adult cohort, we don't really see any extravasation of dye. So that means the blood-brain barrier breaching happens when the animal reach middle age. Next, after peripheral inflammation, we looked at the brain inflammation, particularly in the hippocampus. So here we focused on inflammasome complex, where we see higher expression of inflammasome-associated proteins like NLRP3, ASC. We also looked at the microglial reactivity. So here, across the hippocampal subfields, we could see there are higher activated microglia in all the subfields. But this phenotype does not occur in adulthood. Now, mitochondria and inflammation can go hand in hand. So we looked at mitochondrial biogenesis using Western blotting techniques, and we could see there is a decrease in mitochondrial biogenesis. We also saw mitochondrial dysfunction in this aspect. But these effects were also not present during the adulthood, only they were seen when the animals are middle age. So this is a summary of part 1. I'm not going to repeat that. But the most important part is, can we reverse this? So we thought of a molecule which is a nutritional supplement as well as metabolic modulator, which is nicotinamide. Nicotinamide is an amide form of vitamin B3. It is involved in NAD plus biosynthesis pathway. And NAD plus biosynthesis, NAD plus itself is involved in physiological processes. So we supplemented nicotinamide to these control as well as maternally separated animals for 5 months, starting from 8 months, so that we can chase the cognitive behavior as well. So we could see that if you remember the cognitive decline seen in the maternally separated animals, that was restored in maternally separated animals supplemented with nicotinamide. Whether it is short-term or long-term, the effects were same. Then we could also see the increase GDF15 seen in this case was also mitigated in maternally separated animals receiving nicotinamide. So and other peripheral markers, we could see the similar results. I'm not going to repeat the results because of the lack of time, but I could summarize that if we supplement nicotinamide to these animals, we could rescue peripheral inflammation, we can attenuate the hippocampal microglial activation, there was a protection of blood-brain barrier integrity, and there was a prevention of hippocampal mitochondrial dysfunction. So currently this study is in preview. What next? So with Biju and his team, we would to see whether these findings in our rodent model, whether they hold true in humans as well. So that is an ongoing work. So this is our team. I could not discuss much of our results related to gut microbiome and neurodegeneration, but that work is ongoing with our collaborators. Thank you.
Thank you, Pratik. Now I would like to welcome Dr. Joshua.
Okay, good morning. Thank you to the organizers. Thank you for everyone for having me. I want to change track a bit and talk about sleep. I think we've touched on it lightly during this conference, but I'd like to speak to a chain or a cycle that I've been discovering and researching and kind of getting to grips with over the last 2-3 years. So I'll just briefly touch on some of the data that we found, but I want to make it a bit more broad and holistic to kind of extrapolate that to the BRICS nations. So I'll start by setting the scene. This is from a South African paramedic coming off night shift. Finishes a 24-hour night shift. She hasn't slept. Last night she delivered a baby at gunpoint. She now drives herself home through morning traffic and has to do this again. So these are the types of experiences that our frontline workforces deal with in South Africa with high trauma, high violence. We have roughly 15 to 20% of our workforce doing shifts. This is pretty typical globally. Some data from nurses is that 70% of our nurses will moonlight, which means they work a second job just to have enough money. They either work overtime or secondary nursing jobs. Of these nurses, more than 50% felt too fatigued to work, and 30% of them actually used their sick leave to catch up on sleep. So this is not individual favor, this is a systemic problem. So if we look at the global health crisis around sleep and sleep deprivation, we see that roughly 1 in 3 adults globally have poor sleep or insufficient sleep. And this is more than half, or more than 45% of the population affected globally. Just looking at the US data, we're roughly at $400 billion US. This is now in the trillions of money, of dollars lost to poor sleep. And when we are poor sleep at a basic level, we are 13 times more prone to high error risk. So the emergency medicine workers kind of sit at this intersection. They have the biological consequences of disrupted circadian rhythms. This is circadian misalignment. And they have the psychological weight of high-stakes trauma, violence exposure. So there is a physiological impact in immune function, in cortisol and inflammation, in metabolic dysregulation, and CVS risk. We know this well. In fact, the CDC has put night shift as a carcinogen now officially. We also have the psychological impact, which is on depression and anxiety, PTSD symptoms, burnout, compassion fatigue, and suicide risk, suicide ideation. So why does BRICS bear a heavier burden? Is that I think we're familiar with this, that we have strained systems. We have higher patient numbers. We far exceed the number of practitioners we have to patients. We have extreme shifts that roughly go up to 36 hours or even longer. In South Africa, we run under-resourced when it comes to ambulances, medication, doctors. We have fewer labor regulations, fewer rest period protection, and when it comes to occupational safety and hazard, we are far behind when we compare to, for example, Europe and Australia. In terms of mental health, this is a common thing globally, that is this toughen up kind of initiation, sleep when you're dead, this type of thinking, which is just not the way to go. And most sleep really originates in high-income countries. There's just very little in the LMIC groups. And the existing staff that we do have to handle these high loads without having any backup. And this further compounds sleep loss, and they seek better places because of this. So some of our data on paramedics, this is mixed methods. We use validated questionnaires, we use actigraphy to record sleep, and it's all done in naturalistic settings while they're working. So we found that 71% of them have poor sleep, according to the Pittsburgh Sleep Quality Index. 40% of them are at risk for shift work disorder. This is a DSM-5 diagnosis, and it is excessive daytime sleepiness and chronic insomnia. And when we measured their sleep, we found that they have 3.3 hours of sleep after a night shift, which is some of the lowest numbers we see globally. So this is really quite striking. Just some of the data summaries in terms of the actigraphy-derived sleep metrics. And what we found is that males sleep less than females, but females report subjectively overall worse sleep quality and higher shift work disorder, which means there's a difference between the sexes, and also there's a disconnect between how I think I slept and how I actually sleep, which is part of the problem. So poor sleep and shift work disorder are linked to higher depression symptoms as well. And what we looked at when we compared different shift structures is that the shift structure is not the actual crucial factor, it's the sleep quality that matters. When we look now at emergency room doctors, so this is data we're pulling from 4 public hospitals in the Western Cape of South Africa, and these are dealing with excessive trauma, gunshots, stab wounds. The doctors are under threat, the resources are very low. We found that we again had 71% of them having poor sleep. We looked at 4 different conditions and tried to see how many they each have simultaneously, and what we found is that nearly two-thirds had more than or at least 2 conditions at the same time, and only 17% of them had none of them. So these are the actual stats for that. And what we found is that poor sleep clustered very strongly with depression and shift work disorder. that shift work disorder, again, tracks current sleep. It doesn't matter how many years you've been working for. That's an important difference. And we found that job satisfaction is one of the best things to actually lower the odds of depression, which is obvious, but it's not always a point of focus. So the problem actually starts before the first shift. This is this chain I want to discuss. So sleep education is largely absent from childhood and adolescent criteria, and especially across the BRICS nations, at least for South Africa. So we don't have formal hygiene, sleep hygiene. We have students who use screens, they go to bed late. Chronic sleep debt starts very, very young. I'm sure those of you with kids would know this. Then these kids get to university and they do a medical degree, and they have poor sleep, and they aren't taught about sleep. Globally, we see less than 2 hours of sleep content taught per year. Then they become professionals, and they are already sleep deprived from the university, so they now become sleep deprived in the workforce, and they have this culture of pushing through, which is learned and then taught. So what we never teach, we cannot model, and what we cannot model, we cannot change. So the irony at the heart of medical training is that the professionals we train to heal the body are trained in institutions that deprive their sleep and never teach them why sleep matters. So again, less than 2 hours, 50 to 60% of global university students, especially medical, have poor sleep. Then when they're doing internship in the 3rd, 4th, 5th, 6th years, they have extremely long shifts, underpaid, underlooked after. In South Africa, we had a couple deaths recently in interns, and there's a knowledge gap, which is they don't know much about sleep. And we're actually looking at 8 universities in South Africa now, and we're finding that they do know something about sleep, and they sleep poorly, but they don't seem to care about sleep. This is where the difference comes in. So maybe the importance of it is not truly grounded. So by the time doctors enter the emergency space, they carry years of poor sleep habits, and if they then pursue a professional culture that glorifies exhaustion. And this is the same in the business world as well. So let's talk to the chain, which is a self-reinforcing cycle, unless we disrupt it. So we don't educate our school children from any age roughly about sleep. This continues into university students. This leads to sleep-deprived practitioners. This then affects patients and the doctors. And finally, this culture persists and feedbacks to your children, etc. So breaking the chain, we really need to target the first stage, which is education from as young as we can. And what this might improve is the rate of medical errors that we have in our doctors when they're sleep deprived, the increase in adverse events, and the decreased empathy. So if we look at it, our diagnostic errors increase with fatigue. Medical errors increase on handovers and post-night shifts. Your rate of morbidity and mortality in a hospital goes up during a shift change and over weekends. And the procedural complications, be it surgery, etc., also increase. And there are communication failures that then arise. So our burned out staff, not only are they injuring themselves, misdiagnosing patients, or potentially injuring them, but they start to leave their profession, go abroad, and we have a reduced capacity. Our communities lose our very experienced doctors, and the cycle then continues onwards from there. So let's start education at the school level by integrating sleep hygiene. Let's improve adolescent sleep. Let's include sleep medicine in universities. Let's change overnight course schedules. Let's enforce evidence-based shift rotation limits. Let's look at fatigue monitoring and sleep. And then in terms of BRICS, let's develop LMIC and BRICS-specific occupational health frameworks, because we share a lot of similarities in this. And let's not rely on high-income country data, because it's not applicable. So to summarize, this is about system design. Sleep deprivation is prevalent, measurable, and harmful. The BRICS nations have a disproportionate burden. The root seems to stem upstream in education, culture, and training. Job satisfaction is modifiable, let's use that. And systemic change requires policy, curriculum, and cultural reform. This is my team that are working on the data. I'd like to thank them for all their hard work, and thank you very much for listening. Thank you.
Thank you. Now I would like to invite all the speakers from the scientific session for a 10-minute Q&A. we have any questions from the audience?
Hello. I have a question for Dr. Joshua. So I really agreed with your assessment of what the cohorts and the statistics that you've showed. But I had a question about how much do the genetic factors also contribute to this? Because you mentioned that the education and the systemic effects obviously affect the sleep cycle. But have you had the data available to you to check what are the baseline levels of neurotransmitters in these healthcare providers? Because I only have anecdotal evidence of my healthcare friends telling me that they used to face these sleep disorders even when they were going through med school. And even before med school, I have a lot of friends. And maybe there is a gating of people who need spontaneous dopamine spikes in high pressure situations to kind of function. And maybe it's kind of feeding that system itself also.
Yeah, absolutely. Great question. So I guess I can unpack it. There's the genetics of sleep and sleep disorders. And the people we study, generally, we exclude those with sleep disorders. So this is data from healthy individuals. So there is a propensity, of course, as I mentioned, that so many people have sleep disorders in the first place. The doctors that are with sleep disorders are self-treating themselves, but this is a whole nother aspect of misdiagnosis or underdiagnosis. We talk about apnea, we talk about restless leg syndrome, etc. The dopamine hunting is a very interesting thing. So I'm looking also psychology research in the dark factors. I don't know if you've heard of this. This is sadism, Machiavellism, self-entitlement, vindictiveness, etc. So I'm trying to understand whether doctors are high in these dark factors or low. So are they people who seek this type of behavior, or is it taught or learned as they go along? And it seems not. It seems like they're actually compassionate, empathic people to start with. Whether or not that changes, I don't know.
Yeah, actually, I meant like not in that particular direction, but like spontaneous dopamine kind of helps kind of regulate your basal dopamine if it is low. So if you have more spikes, that kind of regulates your cortisol and your ability to function in a high pressure environment. So these people kind of gate towards those high intensity environments. Now, you're right that looking at the sociological factors like sadism and Machiavellism, again, but that feeds into the systemic effects. And that is very difficult to kind of look at from the population cohort data. You have to look into systemic effects. So yeah, but a really nice talk. I really enjoyed it.
Thank you. Yeah, I'm trying to keep it broad, not go into too much detail, because then we start to lose the message. But I'd love to chat afterwards. Thank you.
Thank you.
Joshua, sorry. Just following up on that previous comment, I completely resonate with what you're saying coming from an hospital-based setting, right? But many of this is very cultural. The fact that you should not, you should not be taking care of yourself, but that sort of always takes a back stand when it comes to sort of helping others. So towards that end, how much of advocacy has already happened in your country towards making the changes that you suggested? And do you think cultural changes can be brought about through these kinds of advocacy efforts?
Yes, I think so. So I agree completely. We in South Africa have a broad variety of cultural backgrounds. A lot of the African groups, sleep is, there's this mysterious or traditional or spiritual nature about it, or it's taboo, for example, and not talked about. In the medical culture, you put the patient first and you work your hardest and sleep. But there's basic things, like having a room to sleep in at the hospital for doctors, bunk beds, for example. Not all hospitals have that, and some cultures at the institutional level don't allow napping on shift, which is just absurd if you think about it. So there's basic changes at that level. So we have private, we have government, and we have volunteer services. I don't know how in India, I assume it's similar. So in private institutions, they should have the full capability at a moment's notice to implement these types of systems. At a private or public institute where we're dealing with government, there's a slower change, but I think the culture matters and the respect, like the same way you don't, we have signs in our hospitals that say don't abuse the practitioners, don't shout at them, don't touch them, that type of thing. Why not have the same type of thing in terms of respect and health for them as well? So if they need to sleep, and this is where comes fatigue monitoring, we should have that type of thing. So that's why I include culture as one of the big ones, because if the culture doesn't acknowledge sleep, we've got a much longer climb before we get there. Thank you.
Hey, sorry, I have a question for Vidita here. So I'm wondering, what is your theory about studying other monoamines in the process of serotonergic psychedelics themselves, considering that the 5-HT2A receptor does interact with other receptors as well, intracellularly and otherwise. So do you think that looking at dopamine or norepinephrine will be worth a shot in this process?
Yeah, people are, because I mean, these, we should, while they're called serotonergic psychedelics, at specific concentrations, they will hit dopaminergic receptors, histamine receptors, noradrenergic receptors as well. So these are dirty drugs. They have multiple hits. And there's quite a bit of interest in looking at the cross interactions between dopamine and norepinephrine in particular in terms of that axis. So absolutely, that's important to do. Yeah. Yeah.
Okay. Hi, Pratik. Very nice talk. I was wondering, when this maternal separation happens and all this systemic inflammation is arising, what is the trigger that starts to build the inflammation? And secondly, do you also see a subpopulation of these pups which are resilient to this? Because in all these chronic social defeat paradigms, the resilient and vulnerable populations segregate. Do you see something similar?
I'll answer the second one first. So we see a spectra on the, so some animals show higher cognition, some animals show they're on the bottom of the cognition. That's why we did the cross correlation. So we do see a spectra, and I believe in humans it will be much more complex that way. The first question was related to onset. Yeah. So although the stress paradigm is in first few weeks of life, so that is a stress hyporesponsive period. So where your glucocorticoids are less, they don't, stress response pathway basically are not quiescent, they are basically quiescent. But so upon stress, body will try to adapt to the situation, and there will be a situation where wear and tear will happen. So that seems to be after mature adulthood in this system. And by middle age, we see a total decline and all the physiological processes in together getting affected, and that results in cognitive aging decline maybe.
So it's for you too. A short question. You show some interesting results showing that early life stress promotes some object recognition memory deaths. I would like to know if you were able to verify some sex difference between in this memory difference, because we are usually see difference and it depends on the age. Yeah. There are some influence of family puberty in this.
So we have in our previous study also, and here also we looked at middle age, adulthood time points. So in adulthood, we don't see any changes in the object recognition memory, if they are males and females. In middle age also, we do see cognitive decline, that is for males as well as females. So there is no sexual dimorphism over there. Yeah.
Pratik, I just wanted to know if we extrapolate it to humans for the maternal separation. So for working mothers, they do get 6 months of initial leave, maternity leave, right? What happens after that before the kid goes to school? So there's a period of around 2.5 years where mom has to go to work, right? So
first of all, we should not directly extrapolate the maternal separation to human. Yeah. So this model represents a model of maternal neglect. So because we have there is a single parental system in rats. So that is one point. We have many other factors surrounding us, the environmental factors, and we have grandmoms, granddads. So those also can negotiate those effects. Only there is abuse or maternal neglect or those kind of things that will impact a child maybe in first 10 years of your life, starting from the 3rd gestation. So that window of P2 to P14 in rodents represents later gestation time till first 8-10 years of your life.
Okay. And Josh, just one small question. So how late is okay to fix the sleep?
Sorry, say again.
Like now that we all realize the sleep is important, So obviously it should start from the schools, or we should be teaching our kids to be sleep conscious. But if people now want to fix, people of different ages like 40s, 50s, 60s anyway sleep more or less, whatever, it doesn't matter. So when an intervention would help to overcome the challenges of sleep deprivation before, is it possible to fix it now?
So you're asking if there's an intervention, is it possible to fix it now?
Yeah, so
Yeah.
somebody who's 45, 50,
Yeah.
if they now get conscious about their sleep, does it help?
Yeah.
Or it's too late?
No, it's never too late. What you do today will carry forward from now. Sleep, they describe it as more like a credit card than a debit card. So you are always in debt, the second you've missed an hour for the rest of your life. But starting now can improve that. And with the shift workers, we see when they're young that they have this poor sleep, which often leads to roughly 10 years shorter lifespan. So the extremity that you do at that age is very severe down the line. But starting from today, you can make improvements in your sleep and sleep hygiene that will then carry on and probably make you healthier and live longer. So it's never too late. Yeah.
Thank you so much, dear speakers, and thank you, audience, for your wonderful questions. You can catch them during refreshments or lunch break or any time to ask them more questions. I'm sure there are. But in interest of time, we will now be starting the scientific session 11. And for this, I would like to welcome Professor Bruno Mota, Professor Biju Vishwanath, and Professor Ekaterina Semina, who would be joining us virtually via recorded talk, but online for question and answer sessions. So, yeah.
So I'm on. Well, so thank you very much. I'm having a great time here. Thank you everyone for organizing and hosting this event. It's been wonderful. And this is going to be a bit of a change of pace. As you can see, I'm a physicist. I'm a head of the experimental mathematics and theoretical biology lab. So in studying the cortex, that's where I'm going to start. So what is theoretical biology? It's essentially creating models. And creating models in biology are not so much about solving the mathematics of the model, but rather asking the right question. That changes dramatically how hard or easy is to solve the model. And for that, since everything in biology is important, it's really about discarding what's important and retaining what's essential. And finding the right abstraction. We're going to abstract something that's very concrete into something that is slightly more abstract, and from there, we're going to extract information that can be applied to the real world again. And to give an outline of what I'm going to do, that's what I'm doing. I have this set of very complex objects, these cortices. I'm going to abstract them first as a movie of a melting cortex, a series of objects that I can analogize to these mathematical abstractions called fractals. And from there, we can express each cortex as a trajectory in this abstract space, and therefore separate different ages, different species, different health conditions. So to do that, we have to ask questions. And I'm going to structure this as a series of questions that I believe have become, I ask myself over the years, and are more and more relevant, more important, more significant to understanding the shape of the cortex. So the first and most immediate question one asks, especially when you think about human cortices, is how does the cortex fold? And although this is not going to be the focus of this talk, I have proposed that the cortex folds like a piece of paper. It crumbles. You have a piece of paper, it has an area, it has a thickness. You crumble it, it generates an object that is folded, it's compact, and it has an external area that you can measure. Now, by measuring these paper balls, you can try to find how they scale and what kind of law expresses the relationship with those quantities. Now, the importance of the paper balls is twofold. First is that it gives us the variables that we're going to be using to express the shape of the cortex. We have, of course, the area of the pile surface area. It's a measure of the size of the cortex. We have the average thickness of the cortex. And once it's crumpled into an object like this, we can measure the exposed area. And that's a measure of extrinsic size of the same cortex. This model that we have created combines essentially two effects, a very simplistic statistical physics model, One is the dynamics of axonal elongation, and the second is the nature of non-interpenetration of cortical surfaces. And the balance between these two, energy and entropy, give rise, after some simplification, to a power law relating the total area, exposed area, and thickness. Empirically, this model has been very successful in predicting the shape of the cortex of various mammalian species in terms of these exponents and in terms of a dimensionless constant called K. Essentially, what this plot here shows is that this constant K is universal across different mammalian species, but not all mammalian species. There was some evidence that cetaceans fold slightly different, and we have recently been able to reconstruct cetacean surfaces in our lab and show that they have a slightly modified cortex. They also obey the universal law, but a different value of K. We think that this is due to the environmental pressure they're subject to. A lot of cetaceans are deep diving. All of them live exclusively on water. And this was the result of this very interesting work of a postdoc of mine that essentially created a network of cetacean collection institutions that we can then collate and scan to generate what's the largest collection in Latin America. And that's the reason why a theoretical physicist has 2 fridges full of dolphin brains in his lab. Now, this is the sort of collaboration that I can also find here in Chennai, and it's essential for the work that I do. This universality in geification has been shown to apply not only to different species, but also to different individuals in the same species, humans specifically, for different regions in the same cortical hemisphere, for different lobes and different subregions, and as we're going to see, for across different length scales, looking at the brains and the different resolutions. Now, this, I believe, is a successful program, but it leads to a more deeper question because it clearly shows that the shapes of all these objects that are apparently very different are, in fact, very similar in some sense, but in what sense specifically? And that leads us to, I think, a more important question, which is, what is the shape of the cortex? It's a question that seems to have an obvious answer, just point to an MRI image and say, there it is. But in fact, it's much more deeper than I thought at first. So to understand this, one have to ask, what does this look like? This looks like an object that has detail at every scale. And if you look at the mathematical formula that relates those morphometrics, those are the typical expressions that describe fractals. Objects that are self-similar, they are similar in small scales as they are in large scales. Small parts of the object are similar to the object itself. And that is true no matter how high the resolution, how deep you go in, how much you zoom in. Now, obviously, fractals are mathematical abstractions, but they can be approximated by objects that are becoming more and more close to them as you include more and more length scales. And they usually expressed in parallel relations, such as the one that describes various cortices. Cortices can be described also in terms of how jagged they are, how much more detail you gain once you go into higher and higher resolutions. So these are the same type of cortices that I showed before, but with different fractal dimensions. The fractal dimensions is as a measure of how much you gain in this scaling. And you can go from something that looks like a line with fractal dimension 1 to something that looks like a filled triangle with fractal dimension of 2. In between, you can have a family of shapes, and it's interesting to see whether the cortex is among one such shapes. Now, this also suggests was the kind of analysis that one must make, because the description I have of the cortex before has a severe limitation. I have this very complex object and describe them as a single point in a 3-dimensional parametric space. Exposed area, total area, and thickness. Now, this is a very broad description. It's not very specific about differentiating my cortex to your cortex or giraffe's cortex. A much more precise, a much more rich description is expressing the human, the chimpanzee, the marmoset, not as point in this 3-dimensional sphere, in 3-dimensional space, but as a trajectory. As I look at the same object at different resolutions, each resolution is going to correspond to one point in this space, and this trajectory is uniquely associated to a shape. And I can then compare different shapes in terms of their trajectories, how they diverge, how they converge, how much they overlap. And that's what we're going to do. But first, we have to find a method of obtaining these trajectories. And what we're going to do is a procedure called coarse grading. We start with a cortical surface, then we're going to progressively erase structures such as sulci and gyri smaller than a certain factor, while preserving the geometric and topological integrity of the cortical surface. We therefore obtain a different surface and can repeat the procedure to obtain further and further degraded degraded structures that have fewer and fewer details all the way to lissencephaly. So I start with the smallest, the small, the medium, the large, and I generate this trajectory, which we're going to see actually overlaps with the universal law that I have found across different species. I won't go into the details about how this algorithm works. I'll just show how this ends up looking like in actual human cortex. So we have gray matter and white matter, and each frame in this movie is the same object seen at a different resolution. So the next frame is going to erase slightly the smallest of the sulci, and then the next frame after that, you're going to erase a sulci that is slightly larger and larger and larger and larger. So can please play the movie? Or can I do this? No. No, I think has to be you. Can you please play this movie? Yeah, so play it again. So you see that the cortex, I call this melting. It's melting of a cortex. It becomes progressively less purified, and if you look at the proportions between gray and white matter, proportionally thicker. Now, this of course is very nice and pretty, but how you quantify this? And this is what you get. We start from the dots that corresponds to different species, a human, a chimpanzee, so on and so forth. And the lines are actually curves that are very straight, that corresponds to the various frames of the various movies. And you see that not only are they straight lines, but the lines overlap. And that's very significant. I can get this for 11 different mammalian species and now a few other cetaceans and non-cetaceans. And what this means is that this universal law not only is valid across species, but the fact that it's a straight line shows that this is a fractal, or a fractal approximation, and moreover, that all of these objects, all of these cortices are approximations of the same archetypical fractal. What changes between them is the range of scales at which this approximation is valid. For a human, the range is broad. For the chimpanzee, it's less broad. For the macaque, it's even narrower. So this is what I call these abstractions that are very illuminating. We can quantify a really powerful and unique universality across various very different looking objects. Now, of course, we know that the cortex cannot fold on forever. It has to be truncated. So cortex are truncated fractals, but what defines the scale of truncation? And what defines this essentially is the thickness of the cortex. The universal law can be reinterpreted as saying that if I look at the smallest features in a cortex, and this is an analogy with a two-dimensional fractal, then the ratio between the area of the smallest feature and the square of its thickness is universal. So it's as if the cortex is made of tiles, and the shapes of these tiles is universal across different species. This is what A0 means. This is the area of the fundamental tile, and it's as long as K is constant, approximately constant across individuals, then it's a proportion to the thickness squared. The ratio between those two things is an aspect ratio, and that's the universality. That's what this universality means. So by looking at this, it makes sense to use to change a different set of variables. And if you think of this three-dimensional space with total exposed area and thickness, this is simply a rotation, where I now find three other orthogonal coordinates that express separately shape and size. And one of them is simply K. I can isolate K, compute K from the total exposed areas and thickness, and essentially say the universal law means that this quantity is approximately constant. I find two other variables, one that expresses isometric size, what's changed if I isometrically grow or shrink an object. Another one that is also dimensionless and also express shape that we can interpret as a morphological complexity. Now, each cortex is then corresponding to a point in this three-dimensional space, the same space, just rotated, and the trajectories also exist in this space. And now we can interpret there in, I think, a very straightforward way. And these variables can be applicable to any fractal. So this is what they mean. Isometric size, what I call I, is essentially what happens is, what changes if I take an object and just increase it in size or decrease it in size. The fractal dimension means that an object becomes more and more convoluted, but if they maintain the aspect ratio of the smallest structure, then it has a constant k. And if I increase the number of degrees of zerification, then I increase S. And what I find is that an object that's straight like this is a fractal of fractal dimension 2.5, or a higher fractal dimension, or not fractal at all, or a different fractal with the same fractal dimension. And different land mammal species all lie along the same line, and humans all lie along the same line, and are remarkably uniform, so that we can really, compared to other species, say that there is a universal trajectory, at least for non-cetacean species. Now, this can be interpreted as the range of scales at which these cortex are fractal, and simply humans have a broader scale, therefore are more gerified. Chimpanzees are slightly less gerified, and the mouse lemur, which is a primate with a smooth brain, is not gerified at all because that's degenerate. But cetaceans are different. Cetaceans have a different trajectory. We only have 2 cetacean species so far, the Sotalia and the Tonia, but they have a different scaling rules, a different fractal dimension, but crucially, they have the same universal fundamental scale. So this universality and this non-universality is what you can get from this abstraction. And just to generalize, just to make an analogy, these are the known cetaceans that we have, and these are analogous to fractal, approximation of the same fractal with the same fractal dimensions. This is the citation that we have, and they analogizes to fractals that are slightly more jagged, but also end up in the same fundamental aspect ratio, the ratio between the smallest structure's size and thickness. And now, in the time that I have, I just want to point out some really interesting results that if I think about developing brains, and that's the work we've been doing here in Chennai with my wonderful collaborators, we could ask ourselves whether a small fetal brain that's not gerified looks like a small monkey brain that's not very gerified, and whether development is essentially going through the evolutionary history of the species until you get to the humans. And we can categorically say that it's not. Different gestational ages have different trajectories that never, ever intercept the adult or the non-human adult trajectory. So that evolution, phylogeny does not recapitulate ontogeny. And this is the sort of question that has been answered in the past. People know that it's not true, but can be now quantified, and we can ask other questions and have them quantified as well. And that's the power of this method. And in the last 30 seconds, I want to go like over a speed run over the history of our cortex over the age. And that's a number of projects that are asymptotically finished. So I'm just going to be very quick about what all they mean. We're going to use K, because it's the invariant in adults, as a probe. And we can see that gerification, given by the gerification index or by the K, is not uniform across development. It really shoots up in humans at about 27 weeks of age. The size doesn't really change the rate of increase very much, but both K and gerification do. So that's one specific instance when cortical circuitry is starting to form we have a start of gerification. Oh, sorry, okay. And if I can have like 5 seconds, this same age is the same age at which premature babies are unable to recover the adult value of K if they are born before 27 weeks of age. So that's a very crucial period. Before then, if it happens in the womb, you will recover the full size value of K. But if you are born before then, that won't happen. We can also find the delineation between development and aging, and the progress of aging throughout the years, both in health and in disease. And I finish by thanking my students in my lab, my collaborators. They are very well motivated, underpaid, but very well fed group of students. And also this wonderful collaboration that I have here. These are some of the people who work directly with me, and you see some of them here working making this event happen, making this event as wonderful as it is. So I dedicated this talk to them, and thank you very much.
Thank you, Professor Bruno. Now I would like to welcome Professor Biju.
Good morning. Okay, slides have to be there. Okay, so I think the slides should hopefully be coming in. So yeah, so I'm a psychiatrist. I work at NIMHANS, and I'm trying to present some snapshots of the work that we've been doing over the last decade with a lot of collaborators, science institutes in Bangalore, and which is a great atmosphere to do science. Yeah, so what we all very well know is that the burden of mental illness in India is very high. So 1 in 7 Indians is affected by a mental disorder. This is 15% of total years lived with disability by human beings. Now, this includes many of the conditions which are listed here, but more importantly, this leads to early mortality, high suicides, medical discrimination because psychiatric patients are not treated very well, and is a very well-known factor in medicine, leading to early death. And so, when I started this kind of work, the primary thing was stigma was a major issue, and I thought that understanding the biology of mental illness will probably reduce the stigma. So, usually I have a set of slides describing many of these psychiatric conditions, but essentially these are, in a snippet, ABC, effects which these affect behavior and cognition. Affect is the emotional state, behavior is what we do, cognition is how we think, and all of these conditions that are listed here actually affect all the ABC of mental health. The important thing to understand is that there is high comorbidity in psychiatric conditions, and treatments are mostly symptomatic, which means there are no disease-modifying medications. Hence, it's important to understand the biology. So what is well known is that most psychiatric illnesses have a very early onset. And even if they don't have they come to the clinic much later, but symptoms are started much, much earlier. So what is currently known, so is that there is a clear genetic basis for many psychiatric conditions, and then there are the environmental factors, the prenatal, postnatal environmental factors, leading to changes in brain plasticity, leading to a vulnerable state in which the person is for many, many years, till the person develops illness. And by the time they come to the clinic, it is very, very late. So to understand this, there are many kinds of studies which are important. One is you need large clinical samples. There has to be study of environmental factors. There has to be study of the brain measures in the live human being, study of genetics, DNA, plasma cells, of course, studies of the brain tissue itself. So I'll just give snapshots of some of these that we're doing. We can take further discussions over coffee. Many large studies in NIMHANS where we are looking at, first, these are studies looking at early development, around 20,000 individuals who are followed up over time from the ages, actually prenatal, to the ages of 30. And of course, there are studies which are looking at people who are adults. And this study specifically is a collaboration with NCBS. We assess patients over 10 to 13 hours with all kinds of assessments, which patients and family members are very kind to spend time with us. So this is around 3,000 people that we've assessed across time, and these people are in follow-up. These are patients with illness and their families. This is a specific study which has expanded beyond NIMHANS into 12 other sites in the country, specifically trying to understand the genetics of bipolar disorder. And here again, we are trying to collect clinical data and samples from 20,000 people. I'll skip this slide. So trying to understand environmental factors, some of them was described by Pratik, trying to understand early adversity, and that's exactly what we try to do in the humans. And here we see that in our sample, 2/3 of the sample actually had adverse childhood experiences. So these are traumatic experiences in childhood, physical abuse, sexual abuse, emotional abuse, very, very traumatic. Now, we do know that this is associated with an earlier onset of illness, and that is what we showed here, but there was also a difference between men and women. Again, the most important adverse childhood event was neglect, and that again was measured by Pratik in his study. What we also know, it's a busy slide, but what we need to understand is that these adverse childhood experiences are highly correlated with suicidality in adulthood. So many of these measures of suicidal ideations, plan attempts are very high in people who have had adverse childhood experiences. Studying brain measures, some of the things that we are trying to study, for example, a colleague of mine, younger colleague, is trying to understand the trajectories of brain MRI parameters. This was again done in a global database. Again, another colleague who is trying to understand the trajectories of cognition, because all of these are not static and this changes across time. Then we try to understand what is happening in patients and their families. And here we, Pavitra's work, showing that many of these are the various domains of cognition that we study. But this is global factor itself is low in many psychiatric conditions. But more importantly, these are abnormal even in unaffected first-degree relatives. So these are family members who do not have an illness. And these people also show many of these abnormalities. So now we're trying to understand whether we are able to stratify these people based on various factors and various biological aspects. When you look at MRIs, again, we find structural changes. So these are all the changes that we see, the yellow, the blue-colored, the red-colored changes these are all areas where there are brain changes. And these are seen in many areas. But these changes are also seen in unaffected first-degree relatives, again, indicating that these are probably vulnerability markers which need to be studied. So how do we understand the correlation between environmental factors and brain measures? So again, one work done by Srinivas, where we try to understand the effect of adverse childhood events on the neurocognitive ability. And there are two groups here, people with a familial risk and people without a familial risk. And we do find a gene-environment interaction here that in people with a familial risk, with an adverse childhood event, the cognitive scores go down. So trying to understand DNA and genetics from these people, Mira has established a lab with around 40,000 samples which we are working with. And here we are not just looking at all these psychiatric conditions, but we are also looking at neurological disorders like Huntington's disease, Parkinson's disease, Alzheimer's dementia. We do know that heritability in psychiatry is high, but most of these are studies done in European populations. So all the blue is European American samples and very, very, very low representation of Asian samples. So what we have tried to do to start with, we have tried to do polygenic scores in our sample using a European database because the Indian database are still developing. But we do find that this is for bipolar disorder. We find that the polygenic scores actually differentiate case and control. So that is something very, very encouraging. I'll skip this slide. We are trying to do similar things for lithium response in bipolar disorder, so not just for the disease, but also to understand drug response. Now, this is whole exome sequencing where people have tried to identify rare damaging variants, mutations in these patients, and we do find many of these, and many of these are also implicated in Mendelian neurodevelopmental conditions. So going forward, postmortem brain studies, here we intend to collaborate with the SGBC in many of these studies. And of course, we have a brain bank of around 300 brains, and we have around many, many patients of schizophrenia and bipolar where the brains are available. Some of them where the brains and the iPSCs are available. So again, this is a student of mine who's here, Vidya, who is trying to understand somatic mosaicism in the scar-12 brain. And this is, again, work in progress. Now, the other major aspect of work that we're trying to do is iPSC-based studies. So we, in collaboration with NCBS, have established an iPSC bank of around 130 induced pluripotent stem cells. Again, in generation, we have around 5,000 PBMCs banked from which we can generate iPSCs. Again, all of these psychiatric conditions are represented. It is possible to convert these iPSCs into neurons, astrocytes, and oligodendrocytes and study them. Initially, we are able to show there are migration deficits in neural stem cells derived from these patients, especially the bipolar disorder. Further on, we have been able to generate with Bhavana and InStem cortical organoids. And further on, we are able to show that this migration deficit leads to reduced neuroepithelial buds and abnormally positioned neurons, again in bipolar disorder. We are able to show there is mitochondrial dysfunction in bipolar disorder, and now we are trying to understand whether some of this is related to the therapeutic effects of lithium and how lithium acts on the brain. Ongoing work by other students in the group trying to understand the effects of lithium in oligodendrocytes, in microglia, and astrocytes, again, some things that we are trying to do to try and understand the biological effects of drugs. It's very important. Most of the drugs we use in psychiatry, we have no idea how they work. And it's very important to study them in human cells. Another kind of work, Maitri is working on OCD, trying to understand the effects of SSRIs. Amrita, who is trying to understand the effects of clozapine, which is a very important drug to try and understand how it works in schizophrenia. So, overall, the hope is that with all these kinds of studies and in collaboration with many other science institutes in the country, we will be able to try to understand a little bit about the biology of mental illness, and hopefully that will reduce the stigma and hopefully lead to better treatments. With that, I would thank all my funders, collaborators, students, many of them are here. Thank you very much.
Thank you. Now can we have Professor Ekaterina's talk online with us?
Dear organizing committee, dear colleagues, first I would like to say thank you for the opportunity to present our results at the conference. Today I would like to discuss the possibilities of using eBiobank to address mental health issues. Genetic factors and the external environment make the main contribution to the development of mental health. These factors are especially important during embryogenesis, where not only brain morphogenesis matters, but also the mother's mental state, her environment and nutrition. All of these can affect the fetus and its brain at the epigenetic level by changing how genes work. It is also clear that many factors act on top of the epigenetic background, and it's impossible to single off just one of them. That's why modern research needs a comprehensive, systematic approach. What is important in biological psychiatry today? We analyzed more than 800,000 published articles on biological psychiatry in the 21st century using a LLM model. On the chart, you can see 10 main topic blocks. The largest one is clinical characterization and biotypes. The next major topics are neuroimaging, psychopharmacotherapy, animal models, and neuroinflammation. There is also growing interest in computational psychiatry, AI, and omics approaches, which integrate large system-level datasets. Such a tool for systematizing large neurobiological and clinical datasets exists at our center. It is the NeuroResource Biobank. Built over 35 years, it now includes more than 16,000 samples. It is officially registered at the National Research Platform and is part of NASBio. A key feature of this biobank is the detailed clinical, phenotypic, and ethnic characterization of the biomaterial. So, returning to embryonic morphogenesis, a clear example of how a large biobank can be used is the study of the link between human accelerated regions or HARS and speech disorders and brain developmental abnormalities. HARS are highly conserved DNA regions that remained unchanged in other mammals, but highly mutated in humans. It is thought that these changes may have contributed to the development of human speech and thinking. Most published studies are based on bioinformatic analysis or on very limited biomaterials, such as cell lines. Our study using biomaterial demonstrates that in children with autism spectrum disorders, ISD, and intellectual disability who also had speech delay, HARS abnormalities were found more than twice as often as in affected children without speech problems. These HAR abnormalities associated with intellectual disability, microcephaly, global developmental delay, and other severe conditions. We identified two large unstable clusters of HARS, one of which is associated with genes responsible for neurodevelopmental process, and the second with genes responsible for cytoskeleton, synaptic plasticity, and microRNAs. Analyzing where these HARS are, we discovered that they are located in enhancers. They do not affect disruptions in the genes themselves, but epigenetically influenced regulatory regions, and such disorders are called enhanceropathies, conditions that lead to disturbances in brain morphogenesis. Delving further into the morphogenesis of the brain as a process whose disturbances lead to the development of mental disorders, we paid attention to one unique participant, the urokinase receptor, UPAR, and its genes, PLAUR. It is part of the urokinase system, which regulates fibrinolysis and inflammation. But recently, results indicate its important contribution to process occurring in the CNS. Normally, in the adult human brain, UPAR is not expressed. However, under pathological conditions, for example, neuroinflammation, toxic brain damage, seizures, its expression hardly increases. In the adult brain, the UPAR gene PLAUR demonstrates immediately early genes properties, and these genes are known to determine the process of neuroplasticity, excitotoxicity, mental and cognitive disorders. In contrast, during embryogenesis, the expression of the PLAUR, UPAR gene PLAUR, increases as the brain forms, whereby the 60th days of the human embryos, it's maximally expressed in the radial glia cells, its neuron precursors, and microglia cells, immune cells of the brain. Expression of the UPAR as protein helps neurons migrate to the outer layers of the cerebral cortex. Here are neurons labeled in green above control and below UPAR overexpressing neurons. And these are precisely the cortical layers that determine cognitive and mental functions. Most interesting of our observation was that UPAR determines the trajectory of axon growth and branching like guidance receptors. Its blocking, here you can see on the right, resulted in disruptions in axons growing, and this solidified our view of the UPAR as an important participant in the mental health, and maybe PLAUR mutation in humans could be associated with mental disorders. So we tested this suggestion using the neuroresource in patients with schizophrenia as one of the most genetically determined mental pathologies. On the animation on the left, study design demonstrates. In addition to studying all the known SNPs of the urokinase system genes, the methodology included polygenic risk scores on neuroinflammation genes. Here you can see on the left PANSS scale scores which reflecting disease severity, including subdomains of positive and negative symptoms and general psychopathology symptoms, as well as environmental factors. Here you can see in the green, complications during pregnancy and childbirth. We demonstrated that the urokinase system SNPs are not associated with the risk of schizophrenia at all, but in combination with neuroinflammation genes, they modify schizophrenia symptoms. The combination of PLAUR gene, for example, RS4760, with pregnancy complication, which refers us to impaired brain morphogenesis, is linked to negative symptoms and therapy resistance. This definitely opens a completely new possibility in diagnostics and gives us the opportunity to personal therapy. For example, if a patient with a family history or with the first episode of schizophrenia has a certain SNP and birth complications, when we know in advance that the risk of development schizophrenia following a negative scenario is high, supplementing the obtained results with in silico approach to assess the pathogenicity of the identified mutations, we can deepen into the etiology of mental disorders. For example, missense mutations, you can see here on GIF or here in the red. Missense mutation in the PLAUR gene RS4760 located in the premembrane region of UPAR protein and may affect its expression in the membrane. Activation of urokinase UPA, which requires the receptor for activity and neuroinflammation response. Finally, I'll show the promise of using serum protein markers of neuroinflammation, neuroplasticity, and the urokinase system to assess severity and and predict therapy efficacy at early stages of mental disorders. In patients with acute STPD, it's schizotypal personality disorder, we evaluated serum markers before and after therapy and focus increase in alpha-1 antitrypsin and elastase NSA, but decreases in GDNF and NOLAS. They decrease in GDNF indicates reduced dopamine neurons protective capacity. The low NSA level suggests there is no active neurodegenerative process. The current conditions is more likely related to impaired neuronal development of function. We observed urokinase decreasing, which plays an important role in regulating hemostasis and inflammation. Possibly its rate of clearance exceeds the rate of synthesis. During treatment, its level tends toward normal, but UPAR shows no changes, which could relate to the Plour RS4760 SNP. Its expression may be in part initially, but elevated VEGF in the acute state indicates clear endothelial dysfunction and likely attempts to repair the vessel wall. So in acute psychotic states, neuronal functioning is disrupted, and these changes affect dopaminergic, vascular, and proteolytic mechanism. Summary, I would like to emphasize that complying a biological portrait of a mental disorders is impossible without taking into account the morphogenesis, genetic factors, external environment, as well as dynamic analysis of markers reflecting neuroinflammation and plasticity. This comprehensive approach allows for an objective assessment of the pathological process underlying mental disorders. Thank you very much for your attention, and I will be happy to answer your questions.
Thank you. Now I would like to welcome our speakers, Professor Biju Vishwanath and Professor Bruno Mota, for a question and answer session. In the interest of time, we'll be keeping it short. We'll take a couple of questions, after which we will break for refreshments, where you can ask them further questions.
Bruno, fascinating talk.
Thanks.
Just thinking, putting on my science fiction hat.
Okay.
Right? So if on the scale of evolution, if things are all fractals and it forms the same slope, if you're now a Martian with 10 times the size of the head, would you now expect the same sort of a fractal behavior, same sort of fractal analysis on their heads as well? Well, you would see more gyrification and stuff in their heads as well as?
In principle, yes. There's no reason to believe that humans, or in fact cetaceans, are the ultimate gyrified brains in the universe. If you have some organism that essentially has the same biochemistry, same cells, same cortical structure as we, they have bigger brains, then the universal law should apply to them as well. So you should have more gyrified species than humans.
Yeah. Are you able to theorize why that particular value of that fractal is now conserved? 2.5 is conserved across evolution. Are there physical constraints that the head can only be this big?
No, my belief, I don't think the skull constraint is a factor in this case. And in fact, I think this is pure physics. Given that you have a certain cortical area and you have a certain cortical thickness, the gyrification follows from the physics. It's not something that has been selected for by evolution.
Sorry, just before we hear the next question, small announcement. Professor Ekaterina has also joined us online for the Q&A session. So if you have any questions, please direct them at her too.
Yeah. Hello, everyone. Sorry, my internet has been blocked all morning, so I can join just right now.
Hello. Hello. You're audible and we can hear you. Thank you.
Hi, my question is for Professor Biju. You have been trying to make some models to look at the molecular changes happening during these disorders seemingly arising very heavily from childhood neglect issues.
No. I think that is, again, these are very, very different parts of work. So we have set of patients where we have assessed for childhood adversity, and that definitely leads to a change in their age of onset. But these people also may have genetic mutations. These people also will also have a genetic risk for mental illness. So when you're doing an IPC-based research, it is very hard for us to model adverse childhood experience in the dish, other than adding cortisol, and there are other methods to go about it. But that is not the same. So what we are trying to do in the dish is trying to only understand what is the effect of genetics mostly. So that is very hard in the dish. So
yeah, I was really wondering if reversing any of these molecular changes can actually benefit, because a lot of these behavioral changes are embedded in the circuits.
Yes. So, which is why we are trying to do organoids and we are trying to do assembloids and all. But then, as you already know, organoids and anything in the IPC-based systems have their own limitations. So it's always a challenge. So that will need the other next level of engineering. Maybe Mohan will come with it. But for now, but the fact that we also have the postmortem brains for some of these patients, hopefully that will help. So we have to look at it from many, many angles. So, yeah.
Yeah. I'm not sure if I'm supposed to be asking, but Bruno, this is for you. If I got this right, the constant offset factor K that you had these plots with K and I believe it was S.
It's a morphological complexity.
Yeah, and you had these very distinct trajectories for different gestational ages.
Yes.
So, I mean, what's the biological event that is doing? Is this something to do with T? Thickness or?
Well, thickness is part of K, so absolutely yes. But I think the most interesting thing that's happening is that those are out of equilibrium states. The cortex is folding and growing so fast that's never at a mechanical equilibrium. So different scales behave at different ways. And you can in fact say that, say, at 23 gestation week cortex is lysencephalic over most scales, but gyrocephalic over a narrow range of scales. So it's a weird way of thinking of it, but if you melt that particular brain, you start from something that's light gyrocephalic to something that's immediately lysencephalic. So you lose this universality, and I think that's a very good indication that this is a highly dynamical process, and equilibrium is only reached after the cortex has time to settle down.
Thank you so much, dear speakers and audience, for your questions. We will now be breaking for refreshments. We will assemble back here by 11:20, after which we will take a group photo with everyone. So I request you to please be back on time. Of the day of the whole symposium. I would like to welcome for this session Dr. Jose Cavalcanti, Dr. Adil Bhagwandin, and Professor Reza, along with Professor Daniel Almeida Fillo. Yes, for the talk. So first, we can begin with Dr. Jose Cavalcanti.
Hi. Hi, good morning, everyone. I would like to thank you to the opportunity to be here. Moreover, I would like to express my grateful Mohan, Risha, all the colleagues that compose the team organization of this event. Thank you so much for the reception. Thank you so much for the hospitality. I really appreciate it. So it's sad, but we are in the last scientific session of this event. For this moment, Professor Adil Bhagwandin and I, we will talk not properly about our technical results, but we were talking about our achievements related by our partnership. Based on this, I will attempt to demonstrate the potential of BRICS collaborative research in comparative neuroanatomy, especially in the context of international partnership. So, Brazil and South Africa partnership is young. I think we have around 3 years of partnership. It started when we were in Shanghai, 2023, in the first BRICS Neuroscience meeting. On that moment, we could identify a lot of convergence of interest of research. 1 year later, when we were in Moscow, the second BRICS Neuroscience Symposium, I had the opportunity to show our first results. And our first research led by Paul, when I take the opportunity to talk about the cytoarchitectonic and neurochemical characterization of subcortical nuclei in rodent brains and comparative analysis. When we're talking about the growth of Brazil and South Africa partnership, it's important to talk that we took the appropriate time in Brazil because we have they stimulate the politics of international collaborations with Brazil. It responds directly. It responded directly to the internationalization priorities set forth by our national research funding agents, which increasingly value global, high-impact effective collaborations and improve our research. Taking on this time, the opportunity time, we could manage funding of 4 different causes of funding of different agents funding in our country. In a few months, was possible to publish some interesting papers. And in this moment, we are working hard in 2 more papers. Professor Paul Menge talks about it yesterday. Moreover, we have the opportunity to launch an important research topic in Frontiers in Neuroanatomy. In this topic, we have the participation of Brazil and South Africa, Professor Adil Bhagwandin, and we have the collaboration of the professor of the China, Zili Huang. He was in the first BRICS Neuroscience meeting with us. But papers, it's important. We know. Everyone knows. But we need to talk beyond the papers. We need to talk about the opportunities for this generation and the next generation of professors, researchers, and students. Based on this, our projects, our funding was possible to manage the opportunities and postdoctoral fellowship. For example, Neliane and Paulo Leonardo, they were in the POS lab last year researching about comparative neuroanatomy. And fellowship, scholarship for visiting professor, Professor Adil Bagwandin, who was with us around 7 months. He was in our lab to developing not only our experiment, our laboratory practice, but our activities and postgraduate program. Moreover, it's possible to generate, have been possible to generate opportunities for undergraduate and graduate students, improve language, improve experience, improve a lot of things. In terms of financial support, until this moment, our projects funded by the call of funders that I talk a little bit the last slide, we have managed around $200,000. It's equivalent in Brazilian currency more than R$1 million. Expand our discussion for the BRICS countries, BRICS plus countries. It's possible to talk. The BRICS plus country encompass about 36% of the world's land surface and 48.5% of the global populations. We have large diversity in cultures, language, and the natural world. About the biodiversity in the BRICS countries, it's possible to say there are over 6,700 species of mammals in the world, over 4,000 species of these animals inhabit in ecosystems found in the BRICS countries. This picture shows us specific numbers of our mammal species around the BRICS countries. It's important to talk that we together, we have more than 63% of mammal species around the world. Based on these numbers, we put in the BRICS Neuroscience Program document, in the final version of the BRICS Neuroscience Program document, the important aim. We aim during the proposed tenure of the BRICS Neuroscience Initiative to collect, section, stain, and digitize the brains of between 5 to 10% of extant vertebrate species. In this moment, I don't talk about mammals. I talk vertebrates. We need to include fish, birds, amphibians, reptiles. In this moment, we are talking about more than 74,000 species. 10% of this, we are talking about more than 7,000 species to collect, stain, section, digitize. It's possible to offer to disposal to the population the biggest collection of brain atlas of the world. So here we have some. If you asked me, it is a very audacious, ambitious objective. But if we are together, it's possible. We can. And then we are motivated by these questions. The 3 questions will guide us the coming years. And to finish, I would like to thank my colleagues in my lab, students, professors, researchers. I would like to thank our agency funding, CNPq, CAPS, and FAPERN. And special thanks our friend Paul Menger. Thank you so much for trust in our ideas. Thank you so much for give us good ideas. This picture is interesting. A simple moment, great ideas. Thank you.
Thank you, Dr. Cavalcanti. Now, can I have Dr. Adil Bhagwandan on the stage?
I'm just going to wait for this to come up. Okay, so Vanakkam, everybody. It's the last session of the day. So I want to continue with what my friend and colleague Jose has. He's put a very nice framework and a landscape of what we're trying to achieve. I'm going to build on that a little bit through this talk. So I've titled it Building with Bricks. We both titled it Building with Bricks. And I don't know how many of you are Pink Floyd fans, but there's a famous song by him called Another Brick in the Wall, and I hope bricks doesn't become another brick in the wall. So with that in mind, I wanted to start off with a quote. I'm sure you all know the individual on the slide, and this particular quote has resonated with me for a number of years. It's become part of what I try to do, and it says, "Be the change you wish to see in the world." With this quote, I hope that what we try to achieve with BRICS, what we try to do with our collaboration, will resonate in this talk and in the future. So, Jose has given you a very good idea of how we came together. I want to reiterate and emphasize that sometimes, much like the nature of science, the simplest things and the best opportunities come from simple curiosity. And that's really what Shanghai was all about. People getting together, neuroscientists getting together, talking about how do we work together, how do we understand what's happening in neuroscience amongst the different BRICS countries, and how do we push that forward for the future. That was elaborated on in 2024 in Moscow. We refined our objectives. And really what we came down to was two rather broad fields, that being the clinical neuroscience and the comparative neuroscience. I think a little bit earlier in the week, on the first day, I mentioned that I'm a comparative neuroanatomist. So this talk will largely focus on comparative neuroanatomy, comparative neuroscience, and how do we take this into the future. So, following up on the questions from Jose, where do we want to go? This might seem like a sensitive topic, but it's important for us to realize some very critical things. The rat and mouse model has been a model that's been used for decades, bordering on, it's been the major model of neuroscience. There are two papers, iconic papers. So this was published by Paul in 2008, and there's a follow-up paper in 2018. And essentially the point of these is that there's been a heavy focus on looking at the rat and mouse and using the rat and mouse to understand brains, and from that information trying to establish understandings about the human brain. I'm not putting down that research. What I'm asking is, why have we not looked at other species? The rat and mouse represent a very tiny percentage of mammals and indeed vertebrates. And there's a great diversity globally. So this is an interesting question for all of us. And I was reminded, and I want to emphasize what Prof. Siva Prakasam had said during the welcome talk when he mentioned that we should consider diversifying strands and creating new ideas and projects. And I think what I'm about to talk about will hopefully address some of what he was talking about. So it's been a predominant model, but in recent years, the translational value of the rat and mouse has come under question. So I think it's time that we reconsider. During the first plenary, I think it was during the first panel discussion, there was a very interesting question from one of our colleagues who asked, given the opportunity that BRICS has at the moment, how do we and how can we use that to further neuroscience? And I think we have that good opportunity now in terms of the comparative aspect to really diversify our understanding, to really investigate more species. And the reason why I'm saying that is because when you look at the biodiversity, as Jose has talked about, Africa and Brazil hold the greatest biodiversity on the planet. We have access, we have resources, we have the ability. We should be pushing this forward. We should be thinking about this. We should be trying to investigate this and develop more and more and further our understandings about how information and ideas from comparative neuroscience, comparative neuroanatomy, can help us further our understandings of the human brain. Indeed, not only just the human brain, but understanding brains and brain evolution in general. So Paul, our esteemed colleague, I'm sure you all know him, he's been championing comparative neuroscience for about 2 decades now. And his collection, although a little bit diminished, is still quite impressive. He has about 210 vertebrate species in brains in his collection. That's a really great starting point. I mean, I think many of you may have already seen some of the papers that have come out from Paul's lab. I was a student of Paul, continued to work with him, and there is so much we've been learning in this time from comparative neuroanatomy. And that's what I was mentioning in the first panel discussion, that we have this wonderful opportunity with BRICS now to really take that a little bit more forward. But building on what Jose had said, Brazil itself has a unique and many unique ecosystems, one of which is the Amazon basin, another which is the Caatinga region, which Karina talked about the other day. There's the Pantanal, and there are iconic species, there are iconic animals, there's a level of, and there's a rich biodiversity in each of these areas. We have that opportunity to do that, and that is what we're trying to build on, that is what we're trying to take forward, and that is how we wish to contribute to neuroscience, moving with the BRICS, in the BRICS arena. So, where do we want to go with this? It's all good and well to talk about there's a lot of animals, and I think somebody had mentioned it a little bit earlier. It's about having the right questions that we want to do. It's just not trying to do animals for the sake of doing animals. But I think an important thing to consider is we need to think about things differently than was done in the past. And one of the ways we can, in the world that we live in, is to create a common language, in a sense. What do I mean by common language? I mean, if we're doing comparative neuroscience, for example, across different countries and continents, we have to standardize the way in which we do this. We can't have people doing different things in different ways. That doesn't make it very comparable. It creates more confusion, and it doesn't lead us anywhere. So one way is to create this platform of a platform and an ability so that we can share information easily. We can discuss ideas, take things forward, even in a simple anatomy sense or neuroanatomy sense. This could be done, and we have the tools and resources. I mean, I think we've all been rather impressed by the kind of capability and the kind of work that's being done in IITM, for example, with digitizing brains and the like. So that's something we would wish to move forward. But one way that we can consider addressing this is by looking at basics, building a solid foundation, using techniques like Nissl, Myelin, descriptive anatomical descriptions, and using simple quantitative techniques. stereology is another popular one. But things like gyrification in his body mass, brain mass, to get general optics of gross morphology and histological anatomy. It would also be a good idea to start creating digital repositories of this. This is in reference to the platform I talked about a little bit earlier. We have colleagues who are already doing that. We saw the beautiful brainstem launch yesterday. I mean, there are incredible things that are happening within BRICS. So I think, again, there are strengths amongst each of us. And if we work together with this, we can really produce something that is accessible to the world in a very different way. Of course, the biggest problem we all have is funding. I was fortunate enough to join Brazil based on CARPS and CMPQ funding, but we have to consider, if you look at South Africa, for example, there is not a lot of money that is available for comparative neuroscience. And that is a problem in itself. So we need to look at alternative avenues to try and obtain money, obtain funding, and use that funding. BRICS would be a very good opportunity in which we could, and I know that is one of the goals to try and raise money for comparative sciences. So we would like to tap into that, should that funding become available. But I also want to impress that neuroscience and comparative neuroscience is not an individual effort. We have to work together to achieve this. And no one person, no one team, no one lab can solve these kind of problems. And this has become a very apparent thing, having spent more time with my collaborator and friend Jose in his lab and his colleagues, about how we can actually establish that and how we can push that forward. So what might the future look like? I like to call it the crystal ball, because by the end of, I'm almost done, by the end of this year, we want to collect about 60 different species from Brazil itself. Jose touched on the 10% of extant vertebrate species that we wish to achieve in future years. But I want to point to something that I refer to as the hub and spoke model. And really the focus, the hub is comparative neuroanatomy, comparative neuroscience. And each of the BRICS and BRICS Plus nations can contribute to that. So we are very keen on people, or looking for people who would like to partner with us to further this venture and adventure of comparative neuroscience. The last thing that I want to just note is that sometimes comparative neuroscience, its value may not be truly appreciated. So one of the goals that we are trying to achieve is to promote that value. With that in mind, I just want to thank, firstly, our hosts for an absolutely incredible symposium. We've been treated like kings in a sense. It's run very smoothly. It's been absolutely fantastic intellectually. So thank you to everybody who's attended. Thank you to the hosts. Thank you for your hospitality. But I also want to make reference to my friend and colleague, mentor, Paul. Thanks for all the ideas during our smoke breaks. You really emphasize that good ideas come from taking fresh air. So I also want to thank my colleague and friend Jose. Thank you for taking the opportunity. Thank you for following up on many different things we talked about. I hope our collaboration will continue into the future and strengthen as we move along. I also want to thank my friend and colleague Karina, who's helped me enormously while I was in the lab in Brazil. Thank you for keeping me sane during those times. So, of course, the students from the lab, there were many of them. They all contributed to, and are contributing to the work that we kind of achieved. So, and of course, the funding agencies. So, yeah, thank you.
Thank you so much. Just a moment of time for some technical difficulties. but I would like to welcome Professor Reza to give his talk.
Okay, I'm the last. First of all, I want to special thanks to all of organizers and Moha and Richa for having me here, specifically in this challenging situation for me in Iran. My talk about NeuroAI. I want to speak about the combination of artificial intelligence and neurosciences. At the first step, I describe a two-way street for you. At the one side, I want to speak about how we apply artificial neural intelligence to discover new things in the huge neurodata. And at the other side, I want to speak about how we get inspired from the brain mechanisms to build algorithm. And finally, I speak about some hybrid computation using the brain organoid. Meanwhile, I speak about the paper that recently we published in my lab in Iran. Okay, at the first step, I want to speak about the primate electrophysiology. We recorded from the monkeys that while observing something and simultaneously from infratemporal network of the brain. And using this data, we want to understand how the external world represented in the brain. At the first things, I want to describe one of my work that did with collaboration with Stanford. We recorded from the monkey while the monkey do delay match to sample task. These tasks include the working memory steps. And we understand that during the working memory, the coordinated activity of two parts of the brain, the frontal and also temporal, can predict the behavior of the monkey. This means that if we have a coordinated activity in trial, this trial have a more chance of successful prediction for the monkey. And as you can see here, when we do some analysis with applying the phase locking value, this means that studying the timing of the two areas, this suggests that the PFC are lead this activity. In the second work, we wanted to address the processing of occluded images. We usually see the faces with occluded object. For example, when you have a mask or glasses on the faces, the brain have a specific mechanism for processing these faces. Again, we recorded from infratemporal networks and understand that PFC are more engaged in the processing of the occluded object, and IT cortex is more engaged in the processing of the intact object. And interestingly, we observed that in the second phase of the response, using some Granger causality analysis, we observed that there is a feedback from the frontal cortex to the temporal cortex that mediate the temporal cortex for resolving the occluded processing. Also, we observed that when the monkey are engaged in occlusion processing, there is more synchronized activity in the infratemporal network. Okay, till now I speak about the monkey neural data. In the second class of the work that we did in our lab, we recorded from the human using a high-density EEG data. And in one work that we used the similar stimulus, the occluded stimulus, and recorded from the human EEG, we found that when we use the similar stimulus, one time labeled as an occlusion type, and another time labeled as an identity. As you can observe, there is a late processing in the occlusion processing in the brain that is completely matched with what I see in the monkeys as described in the previous slide. we developed some techniques that compare different domains using the AI techniques. One domain comes from the EEG, another comes from the monkey primate electrophysiology, and another one comes from high-ended neural networks that we used for the image processing. We made a common space that we name it resemblance similarity analysis. And by comparing these matrices, we understand that the current models only can consider the first stage of the processing in both monkey and human. And the latest stage processing that contain the feedbacks doesn't consider in the current state-of-the-art networks. Okay. In the next word, we use more AI stuff for understanding the brain. We establish a closed-loop systems that simultaneously, in the real-time regime, we get the response of the brain and use some genetic algorithm in some encoding space to produce the good stimulus for the one specific cells. As you can see here, the monkey observed, we started with some noisy stimulus. As you can see here, the evolution of some of them. And the response of the neural activity of this part of the brain provides some input for the AI algorithms to generate a new stimulus. This is a really integrated AI and neuroscience techniques for understanding the brain, and it's in progress work with Harvard collaboration with one of my students in the Harvard. So till now, I describe how we can use the AI for describing the neural data. At the other hand, the mechanism of the processing in the brain is a very good thing for getting, very good thing for, Can I stay in the previous slide? Okay. Good things for getting inspiration for build the model. In this model, we use the one fact in the brain that there is two pathway of the processing, where and what pathway, and apply this knowledge for making a split fuse transformer machine for processing of the video. And the final part of my speak is about applying some brain tissue using a genetic manipulation techniques to provide some primary culture that can come from the mouse rat or maybe come from the human brain that we name it brain organoid. We culture the brain organoid on some silicon that can capture more than 4,000 channels of the recordings. So we have a hybrid silicon and carbon processing systems that we could record a lot of information. You can see some recorded. This is the response of the brain culture in some sample channels. And we wanted to use this culture as a hardware for implemented AI algorithm on it. There is one problem for current AI algorithm, that they use huge amount of energy. Thank you. And if we can use the brain organoid as one thing that's capable of represent the AI algorithm, it is very interesting things. We try to implement the reinforcement algorithm on the brain organoid in some fashion. We have a Pong. This is some actually play that we encoded the environment on this culture and using one places as a motor cortex to give the command to this play. And after a while, using the some regime for the training that use free energy regime, we can build a culture that play after a while. You can see here, we get the command for moving right and left this bar from these two patch. This is the activity of the neural organoid. This is the sensory channels. And now we have a desynchronized pattern as a punishment and have synchronized pattern as a reward. After a while, this primary culture learned to play correct with less than 1 watt power compared to the computer. And you can see here at different trial when we have closed loop or correct training. Yeah, this is the learning curve. This is really good. And when we don't have any stimulation, this is not a good training here. Thank you very much. We are open to collaboration, and I hope it was great. This is all my collaborators. Thank you very much.
Thank you so much. No, we'll have another talk. So now I would like to welcome Professor Daniel Almeida Fellow, and his will be a 15-minute talk as he was the featured presenter. Thank you.
Hello, everyone. Good afternoon already, right? Sorry for making you wait a little bit more for the lunch. But I needed to come and discuss a little bit with you about what I have been doing and in our lab and what I did during my postdoc. So the idea is like the discussion that I need to have with you today is about how can we model cognitive optimization from memory linking mechanisms. So I'm going to try to explain to you what it means, memory linking, and how can you update that. But first, I want to start about the function of the brain. So everybody knows here what the brain is, what the brain does, and everything. But the main function of the brain we can summarize as the main organ that makes us communicate and interact with the world. So the idea is that we have our sensory systems, we have the processing in the brain, and then we have the motor output with the world. So we have this cycle running on and on several times. The whole idea here is that the definition of cognition is every mental process that happens in the brain directed by some sensorial input or internal representations, okay? So this is cognition. But the main important thing here is that, as Heraclitus said, no man ever steps in the same river twice. So even though we may have the same stimulus sometimes, the brain that faces this same stimulus is not the same. Every time we get some new information, the brain changes to accommodate, to interact, to integrate this new information, and it changes a little bit, and a new motor output or a mental representation, and then you go and have these several iterations of information process. But the problem is that the brain is has a finite space. So we cannot include information forever in the brain, right? So the brain is plastic. Everybody knows about that. So we need to include new information here. Every change that is caused by sensorial input can be regarded as memory, or the engram. The physical substrate would be the engram, but the mental representation would be memory. But we need this stability plasticity dilemma. We cannot change too much the brain until you disrupt previous memories, but you need to change sufficiently so as to keep the information that is relevant, right? So we need this stability plasticity dilemma. And I'm going to make a comparison with the computer, for example. In a computer, you have the memory of the computer. You buy a hard drive, and you input information, a lot of information, which is virtually infinite, right? You can always buy more hard drives, more hard drives, more hard drives. But you cannot do this with the brain. Okay? So what do you do when you have to include new information in a subset of already integrated information without disrupting them? What you do is like you sometimes use the same representations, or at least representations that are very similar to what you already know. Let me give you an example. Let's say you have a picture of your marriage, and you took this picture, and you put this picture in your computer, the hard drive of your computer. If you open this picture 10 days from now, the picture will be exactly the same, right? So you see everything the same, but the opening that picture does not increase the likelihood of opening other pictures of other marriages. But with the brain, this happens. If you go to a marriage right now, you're going to remember a lot of other marriages that you went before. This is because the similarities are recorded in the same subset of neurons. So this is called memory linking. The retrieval of one memory increases the likelihood of retrieving the other. And this can be by a conceptual linking, like the one I just said, with the marriage, the concept of marriage, but can be also in time. So when you see some catastrophic event, when we experience some catastrophic event, usually we remember everything that happened during that specific day, right? Several days from now, you remember everything that happened because that specific memory increases the likelihood of remembering the rest. So how do you do this experimentally? How do you test this in animals? So the idea that we have is like, for example, you have the animal exploring one context A, 7 days later, one context B, 5 hours later, context C, and you give an immediate shock in C. So what you expect, usually, when you're going to test this, is that this animal is going to, okay, I remember the place I was shocked, C. I will not remember A and B, right? That's what we think would happen. But this is not what happens. What happens is that you don't remember the object, the context that you experienced 7 days before, but you link B and C because they are close in time. So this is memory linking in time, right? So you are linking two memories that are connected in time. So the animals freeze when they are in C, the animals freeze when they are in B, when they are tested in B, but they don't freeze when they are sufficiently freeze when they are in A. So this is how we test this. And then we went to understand how can we bring that to a representation of the neuronal activity of what's happening here. What's the mechanism of memory linking regarding the neurons? So since I read like Donald Oldingham, The Organization of Behavior, most of you must know that I always dedicated to understand how any of the behaviors that we do, any representations that we have, are represented in the neuronal activity, okay? So in here, what is the hypothesis is that when you are in context A, you activate a subset of cells, and these cells, if these cells get so active that they have increased excitability, and then in episode B, which is very close in time, you use the same subset of cells to record also B. And then when we remember B, you also remember A. When you remember A, you also remember B. So they become linked, but not with Z, which is far in time. So, and this we look where? In the hippocampus, dorsal C1, which is known for contextual information processing. And this, what's the molecular mechanism of this? The hypothesis was, okay, we know that after we have an experience, we increase CREB. CREB increases the excitability, and these neurons with high excitability, they are recruited for the next memory. And this is what we showed, that the neurons that are recruited for the first memory, they increase excitability during the next 5 hours. So any other memory that gets there after this
they are going to use the same neurons to record the same thing. And the hypothesis is also demonstrated experimentally. When we record the neurons in the dorsal C1, we can see that the neurons, the overlap between the ensembles that are active in C and B is higher than the ones that are both active in C and A. So the overlaps is bigger. So what about the dynamics? It's not only the activity of the neurons. So the neurons are active or inactive. It's not the on and off. It's like, what's the dynamics of the neuroactivity? So this is something that we investigated using the UCLA miniscopes developed by Daniel Aharoni at UCLA. And the idea is like use these micro lenses and calcium imaging with the miniscopes, 3 grams on the head of the mice. And then we can use this with animals freely behaving. So let me put again the video. So the mouse is freely behaving, and we record the activity of the neurons. Essentially what we do is like we make the neurons become fluorescent when they are active. And then we record a lot of neurons. So what are the advantages of this in comparison to microelectrodes? They track the same cells across days and months, record large neuronal populations. I recorded 1,092 one time. We can identify the neuronal types, if excitatory or inhibitory neurons. And the drawback is that you have a very low to middle temporal resolution. Another thing that I did in the lab is like to optimize how we calculate and identify these neurons computationally. So previously people were doing, you have different sessions, and you need to know what's happening in each session and follow the activity of the neuron across sessions. So what we do is like get the videos, detect the cells, and then align. What I did is like, I created this concatenation pipeline. So the idea is that you put the videos together, create a big data, and then you analyze and follow the same neurons across time. So this is something that improved cell detection, contour, neuron activity, and neuron activity separation. But what would be the hypothesis of what is happening in the dynamics of the neuron? First, this is what we showed, overlap size. So essentially, when you have linking, the overlap is higher. When you don't have, the overlap is lower. But you have those purple cells there that are overlapping. But we also may have the increased stability of neuron activity. So within that subset of purple cells, you may have cells that are equally active in both. They are more active in one or more active in the other. Okay? So this is the stability of neuron activity. And we also may have another level for that, is the stability of coactivity. So we have these 2 cells. You can see very clearly that these 2 cells, they fire together. Okay? They fire when one fires, the other one also fires. And then when you look at context A and context B, they are firing together in both. So you have the stability of neuron activity. So we have 3 levels of dynamics of neuron activity that you can look into. So what we did is like, we look at the regulatory mechanisms of memory linking, the modulatory mechanism of memory linking, and the disambiguation mechanism of memory linking. How can you separate when these contexts are different, when these contexts are the same? So, and what we did is like, we published this paper showing that once CRAB increases the window, the likelihood of using the same cells to encode 2 different memories, we have to turn off the system. Otherwise, we're going to link everything, right? How do we turn this off? How do we regulate this? CCR5. CCR5 is a receptor used for the infection of HIV. And we found that when we block CCR5 in a 2-day set, so 2 days difference between context A and context B. Remember, they linked when it was 5 hours. 2 days, they don't link. So in the controls, we don't link. But when we inhibit this, we have linking. So these animals, the window is not turned off, so they link after 2 days. But the problem is that in the 2 days, we don't have sufficient difference in overlap between this. So we had to look in a different level, not the overlap size, but the neuronal activity. When you look at the neuronal activity, we see that compared to wild type, the knockout mice, CCR5 mice, they remain after 2 days with increased activity. So the stability of neuronal activity is higher, and then we remain with that. In the neuromodulatory mechanisms, we found that the locus coeruleus to the dorsal C1 circuit boosts this memory linking. So we tracked the projections from the locus coeruleus to the dorsal C1, and we showed that if we inhibit this, even in the 5-hour linking, we can disrupt memory linking, okay? They should link in 5 hours, but they don't link because we disrupted it. And also, we showed that these neuromodulatory mechanisms also show the overlap and also show coactivity stability, okay? And finally, the disambiguation. So, okay, 5 hours, they link to different memories. What if these memories are similar or the same? If the animal is exploring the same context, how can we tell them apart? Okay, we have to disambiguate these memories. How do you disambiguate this? So we investigated the role of the prefrontal cortex on this, because the prefrontal cortex is known for encoding the long-term memory. And we have investigated the cells using the miniscopes, the cells in the prefrontal cortex, 7 days or 5 hours apart in a different context setup or the same context setup. What they show is that the different context setup, we have an increase in activity after 7 days, and for the same context, it decreases. Just finishing. So what happens here is that the prefrontal cortex is signaling the difference. And what happens if we look into the linking mechanisms? What we showed is that we investigated all the pathways from the prefrontal cortex to the hippocampus, so the direct and indirect pathways, the medial interneural cortex, lateral interneural cortex, the dorsal hippocampus. And we showed that the medial interneural cortex is the one that drives this action of the medial prefrontal cortex towards the hippocampus to drive memory linking. We also showed that the overlap size happens here, is modulated by this pathway, and also the stability of neuronal activity. So we also showed that other regions involved in contextual fear memory, they are also involved, they have the same mechanism. So this is the retrosplenial cortex, used here the TET-tag system to show that we can induce memory linking even if a 2 days interval, if we reactivate the same cells that were active in the first context. And we show that these also show overlap size, stability of neuronal activity, even using a decoder, and stability of neuronal coactivity. So essentially, we show regulatory mechanisms, how this turns on, how this turns off, how we modulate this, how we make it stronger or weaker, how can we disambiguate what are different contexts from same context, and we show that this seems to be a more general mechanism. So these results highlight the mechanism related to the integration of a new episodic memory within a cluster of previously stored memories. The results also suggest that this mechanism might be general mechanism across brain regions, so hippocampus, retrosplenial cortex. We also showed this in the prefrontal cortex. Being active is not sufficient for a neuron to be part of a neuronal engram, and the dynamics of neural activity adds information to the proper integration of episodic memories. And finally, these results may help the development of computational models that can help upgrade artificial neural networks that give rise to AI, and most importantly, help us optimize human cognition and face challenging cognitive deficits related to aging, dementia, and neurodevelopmental disorders. Okay? That's it. Thank you.
Thank you so much, Professor Philo. Now I would like to invite Professor Reza, Dr. Bhagwandan, and Dr. Cavalcanti for a combined Q&A session for, we will take a couple of questions, and in the interest of time, and we will get the rest during lunch and refreshments.
Thanks. So this is for Danielle. Great talk. So if you have an engram for one memory, and another engram from another memory, and the engrams are too similar, presumably one can delete the other. And if that's so, can you use this to perhaps estimate the size of a memory in a brain or in a brain region?
So it does not erase the other, but it's the idea is that you have a so much complex subset of neurons, and they are spread across different regions. So even if you use in the hippocampus the same subset of neurons, but it's not all the same. You have, for example, let's say 100 neurons, okay? So 100 neurons that are active during context A, 100 neurons that are active during context B. You have 30%, for example, that it is active in both. You still have the other 70% in each one that can separate them, right? And these other, the separation neurons, like we showed this with more detail in the paper, they are driven by the activity of the medial prefrontal cortex. So this is what drives the differentiation between the memories. So you have a subset that makes you one remember the other, but you have other subsets that help you differentiate both. And about the size of the memory, we have tested this with contextual fear conditional, social preference of food preference, social preference, and so many other behavioral paradigms. And the size of the engram is usually very similar. It doesn't change very much regarding the different types of experiences.
Hi. My question is for Mohammed Reza. Thanks for the great talk. But towards the end of your slide, I'm only starting to understand these paradigms. But were you talking about cells responding to reward and punishment in terms of, was it an approach avoidance paradigm?
Okay.
Could you, that was very intriguing. Could you explain that, please?
It's a tricky question, good question. When we have a culture, for training the culture, we have different paradigm. You know, there is a rule that we name it Hebbian. When the neurons fire together, wire together. I describe some brain culture that the activity can lead to the one action in the environment, in the gaming environment, okay? If the action was good, we imply a synchronized signal to the brain culture. So the synchronized culture induce the simultaneously firing together activity, okay? So the activity that lead to the correct response emphasizes by the synchronized activity. And if you have a wrong answer, you imply a asynchronized stimulation that say that this activity and this connection not good for such playing. This is the regime that we name it free energy. If you know that there is some principle for brain coding, we name it predictive coding, that all of the natural system tend to go to the lower entropy systems. And by this mentality, we use this free energy system for implying the Hebbian rule.
Thank you so much. Thank you so much, dear speakers and the audience, for your questions. If you have any more, you can please catch them during lunch and refreshments so that you can ask your questions. Now, we will be ending our scientific talks for the symposium, and we will be having a panel discussion on building and strengthening multidisciplinary neuroscience research. For this panel, I would like to welcome Professor Jose Cavalcanti, Professor Mohana Shankar Shivaprakasam, Professor Vidita Vaidya, Dr. Joshua Davimes, and Dr. Ileka Malungo. Can we please have you on the stage? And this will be moderated by Professor Pamela Melano-Carpus. Thank you.
So it's a pleasure to welcome you to this last panel discussion addressing on building and strengthening collaborative multidisciplinary neuroscience research. So neuroscience is maybe the most inherently multidisciplinary research field we saw along this symposium. We collaborate between different disciplines, include medicine, physiology, biology, psychiatry, physics, and others. So I would like to initiate this panel to begin inviting each of our panelists to share an example of how multidisciplinary collaboration has influenced your own research on or trajectory and career. Anyone could start, if you like, Ilk.
Yeah. Hi. So it's basically taking off from Prof. Bhagwadin's research, in the sense that the comparative neuroanatomy has really built a strong foundation for my knowledge of neuroanatomy, and then being able to apply that in other kinds of circumstances. So I've kind of veered now into a bit of neurodevelopmental neuroanatomy, but then it's also being exposed to other, like you correctly said, other kind of disciplines. I think we've also started a fellowship at Wits University, in which we are trying to collaborate with a multitude of different faculties. Because I think, universities are just inherently structured in different faculties, and it's very hard for, them to come together. So by doing those or creating those spaces, I think that is where we can, really strengthen, the multidisciplinary.
When you try to work across scales in neuroscience, and neuroscience seems to have this particular challenge because the scales are quite extensive, not that they aren't for other organ systems and tissue systems. I think starting from the very molecular sort of studies where now large-scale data sets, especially when one uses omics approaches, one's dealing with very large data sets. The other end, you're also dealing with large data sets. So I think there's no question that there's a vital need for crosstalk with people who have some degree of comfort with this. So multidisciplinarity is sort of inherently inbuilt. I think the challenge really comes from people tending to look at if I go to Paul Manger's example of the elephant brain in a sense, this is like each of us is looking at that problem with the lens that we have, and the lens often tends to be very limited. And so you end up with a very incomplete picture, and this is the big challenge in neuroscience. I just wanted to add, if I can, one more thing, which is the panel discussion is about what could we do to encourage multidisciplinary collaborations. I wonder if there's a possibility to come up with something like the Human Frontiers of Science Program Fellowship for BRICS. Something that allows for really talented people who are just finishing their PhD, doing their postdoc, and can move across BRICS nations to allow to facilitate these kinds of multidisciplinary collaborations. I think that's going to be vital because none of us was trained originally to be very easily able to move out of the silos. And I think the next generation needs to have that taken away as early as is possible.
Thank you. So I think the interesting aspect of multidisciplinary research is how we define it, firstly. So, I mean, within neurosciences, you have such diversity. So imagine doing your PhD in neuroanatomy and then doing it in music, for example. It's two different things entirely as compared to, that's still under the sciences. Then you can cross over to engineering, pharmacology or medicine, etc. And it's not just a different language that we have between two disciplines, but it's actually different ways of thinking entirely. So the same way that the BRICS nations, we all speak a different language entirely, but our brains differ culturally, and our training changes the way our brains function as well. So we were discussing this yesterday, that it's not just a language barrier, but there's a brain functioning barrier. And I think the biggest challenge when it comes to multidisciplinary work is actually just taking the first step. So we all kind of have this imposter syndrome, I would think, on a daily basis as a scientist. I don't think if you don't have it, you aren't anxious or curious to learn. And I think that's important. But just reaching out to people as far away from what you normally do is one of the best things you can do. And I would always recommend having an engineer in the team because they truly are open to things. They don't understand no, and they just don't, you don't block the way they think, they let them do their own thing. And mathematicians, I've worked with them in the past from South Korea, very interesting group, different way of thinking entirely. And then branch within your own fields as well. Being human means you should understand psychology and psychiatry to some degree. And often what you practice, you should preach, which means if you're experimenting or studying something, you should involve and go deep into that practice as well. Otherwise, you're studying it from an objective perspective, truly, which is good for science, but it also gives you a lack of depth. So the multidisciplinary framework, I think, has a lot of dimensions to it. It's not a very simple, straightforward thing.
So, see, actually, if you really look at it from a very practical sense, not just neuroscience, anything complex cannot be done without at least few disciplines. Right? I mean, it's sort of natural. Because neuroscience was part in biology, naturally earlier, it just became is biology's remit sort of a thing. But if you look at, let's say, something like cardiology, not too far from the brain, even very early days, you had physics, mechanics, not so much chemical, and electrical, because it had, not that the brain doesn't have electrical activity. But it's very interesting how initial conditions set the tone for how a field evolves. Very much like how humans are, right? Now, I always wondered, this is not to put anybody down, I never actually understood this need for interdisciplinary, need for multidisciplinary. If anything is complex, right? So, the way, I don't want to give you an abstraction or theory, I'll just tell you how, we are able to do this. I've always found if you set a mission, and the mission doesn't have to be like a heroic mission, right? It could be, it should be bigger than a lab question or a lab problem, right? Then there can be multiple questions and problems which require people to graduate PhDs, which means, you have to sort of defend your turf or territory. That's okay, because, when you're young, you cannot be asked to think interdisciplinary. That's too much. They have to be territorial so that they can get their publications, because that's the system that we designed. Right? There's nothing you cannot change the system, right? You cannot go and get you cannot defend an interdisciplinary thinking in a PhD defense. You got to start showing. But that does not mean that one does not get trained in an interdisciplinary way of thinking. Right? Your work can be in biology, your engineering or whatnot. So one of the ways, and I steal this from idea is what you had said. If you set up a larger mission, right, then people are also comfortable. Right? And inherently a larger mission like, what I am heading now, anyway you need multiple disciplines. Right? So from a BRICS standpoint, I would say if you set a larger, which inevitably becomes a broader mission, it will become interdisciplinary. Right? And the individual labs can have one discipline, two disciplines, and you will end up collaborating, and then you will automatically start speaking the language of multiple disciplines. Right? So that's all I wanted to say.
So regarding the multidisciplinary collaboration, I have a simple example of my career today in comparative neuroanatomy. For many years, I was primarily interested to describe brain structures across the rodent species. But collaboration with colleagues from zoology, ecology, physiology science, computational science, we are able to transform anatomical perceptions, include questions about brain function and evolution, for example. These interactions expand both the scope and the impact of my research.
Hey, thank you all the speakers for your valuable insights and for share your examples. So some of you, Professor Vidita mentioned a suggestion about a BRICS program promoted is multidisciplinary collaboration across the countries. Thinking about that and looking for the universities' structures and from funding agents, we know that they are still organized around traditional disciplines. So what institutional change do you think that would help to foster this multidisciplinary collaboration, not only on neuroscience, but in science in general? Anyone could give some insights.
So it's, I'm going to use the example of the Human Frontiers of Science program only because there's an interesting idea there, and it may become a potential model from which we can draw for this sort of a BRICS multidisciplinary. If you have long-term mobility, if you have 5-year mobility grants that allow people to cross across when there's a broad project in mind, and the funding is dedicated to mobility, which is to let young people travel, stay, work, and allow and facilitate that, and that becomes a dedicated funding pool that people competitively apply to. There are often these bilateral collaborations between countries, etc., but they don't very often necessarily go anywhere. They get signed, but they don't necessarily result in anything. But if you have it run the way the HFSP is, which is that they're given the pot of money, it's independent of each country in a sense, and it's run as a collective enterprise to allow for this sort of movement, in this case of postdoctoral fellows or young early career scientists. It actually gives something tangible. My worry becomes that when it's something just signed on paper and there isn't something tangible, it can become a bit of a challenge. So I would say starting small sometimes is a good way to go.
Yeah, sure. No?
I agree. I don't have much to add on this, right? Because it's better to walk backwards when you're trying to create a change, which is you measure, let's say, let's take this group, right? 50 people are here. I would measure whatever program that you're going to create, I would measure in 5 years, maybe we'll have a review at 3 years. How many, and we can measure this quite easily, right? How many months has the person spent in another lab, preferably the one that they already were not collaborating with, right? It's an easy measure. Right? And if that's what you want to achieve, right? you can enable it with travel grants, right? So it's the measure that is important. See, many times when I hear phrases, yes, you can't have quantitative phrases as targets, right, or titles, but your measurement has to be quantitative, right? Because you can't get into people's head and said, oh, is this guy interdisciplinary? Right? That's it's very difficult to measure, but you can measure it through so many direct and proxy measures. So if you set that as the goal, then people will apply. I mean, essentially, people follow where the funding is. Even if the good ideas, if they don't get funded, they don't get funded. So I agree with Vidita on this. You measure the interdisciplinary outcome, right? Publications and everything come later, but in fact, publications, I would say, it's a weak measure, right? If you're going to, if your objective is to generate interdisciplinary thinking and multidisciplinary labs, you just measure that directly. So that's what I would say.
So thank you. So we will have about 5 minutes. Are you open to the audience if you have any question for our panelists? We make this session short because the time, just so you know. Yeah.
I think everybody is hungry. Very clear. Lunch. It was a long day.
So please forgive my ignorance. I think there is adequate data from rodent experiments. I sincerely request all of you to move to more translational research, more human-centric research, whatever limited resources we have, including in terms of time and energy. No need to just extrapolate whatever is happening. Get the real human data. I think there are few collaborations have been mentioned last 2-3 days, but focus more and if you do that, even clinicians will be part of this work. Thank you.
You'd like to comment? Yeah.
As a neurosurgeon, I can completely relate to what you're saying, right? Because my core is I develop healthcare technologies, medical technologies. So if you want clinicians, so since we talked about interdisciplinary, one of the disciplines is medicine broadly. If you want clinicians and surgeons and physicians to work with you, you got to give them something which at least a promise in their mind that it'll get into patients. I mean, even if it doesn't get into patients, it shows a certain path. Now, I'll give you a counter to that because I live 2 lives practically, right? I mean, I live the medtech life and then I live the neuroscience life. In fields like neuroscience, you need to advance, or rather, it is still not advanced enough to have this translation in a critical mass way yet. Right? It will take some more time. And this is something that I realized that a while ago, and more so after the last 4-5 years, there's enough needs to be done on the basic science. Right? And whether the basic science is the animal model or human model, that depends on the context. Right? But it needs to move enough before the clinicians can play an active collaborative role like oncology, or medical devices, right? And various other fields. So this field needs more work before this becomes a natural linkage. Right? But because we are talking about humans, I'll put the ball back in your ilk's court, is the inputs, be it specimens, be it clinical material, right, has to come from you. Right? Because that is not something that can be simulated or emulated in the academic labs right now. Right? So there, I would say it's a little bit weak on the clinical side. right? Especially in, I would say, BRICS countries.
Thank you.
So just to build on that idea, because policy decision makers often tend to have such a strong emphasis on a translational outcome that the risk is that if you do not allow for the possibility of a rich ecosystem in which fundamental research can exist and thrive, that you are likely to hamper the possibility of translational outcomes. And the worry with that is that the desire is to have the, whatever, the silver bullet or the perfect solution right away, but you haven't taken the time to do the work, to do the building blocks. So my worry with that is only this pressure on policymakers to shift funding entirely towards translational outcomes is likely to jeopardize the entire enterprise and harm the entire scientific biomedical ecosystem. This is not to say that you don't need the continuum. You need the preclinical, clinical continuum, but if policy gets shifted so strongly towards give me an output tomorrow, you may actually jeopardize the entire enterprise in its entirety. So I'd worry about that, actually.
Just more one.
I'll just add to what Vidita said. You know, if we talk about India, we are like currently full steam. We have brought in so many bright brains back to the country with the promise that they can have their own lab and freedom to do research. And suddenly we say, no, we have changed our goalposts and you have to work for a product. But even for the product, as Vidita said, the biomarkers which we are using for cancer biology, the CAR therapy, and the other things are all outcomes of basic research and animal research. So we have to strike a balance between both. The policymakers should involve basic researchers and clinicians to decide a ratio, let's say 40-60, 20-80, whatever, but it cannot, the basic research cannot be shut down. At least at this point. We should not make that mistake, and I think I would request all BRIC countries to go back, and if you have a similar push, please make them understand, advocate for basic research. That's all I have to say.
Thank you. So I would like to thank you all the panelists that we have in this afternoon. So I hope that today's conversation inspire new multidisciplinary collaboration. We are in a place that is a beautiful example of how different disciplines can work together and build meaningful results, meaningful products that are sharing with all the world. So hope you enjoyed this panel, and thank you again for all the participants.
Sorry, what did you say? You're saying hold it and they will come.
Mohan sir. Closing remarks.
So, we are towards the end of the program. I mean, there's a visit to a 7th or a 9th century monument, 9th or 12th, I forget. So before that, let me just, because we may not be all in the same room and the bus. So first of all, I want to thank everybody, as I said, to take your time out to come here. It's not easy to take a 40-hour flight. So first of all, I want to thank you. And organizing this has been a really easy task for me, because I was just reflecting back. I actually have not done much, right? It may appear that I have done a lot, but all I've been doing is signing, right? Or forwarding some emails, which they ask me to send it to somebody. Sometimes I forward it to the wrong address. Right? Practically all this work has been done by Richa, Kritika, and their committee. So it's very hard for even me to sort of truly picture the kind of complexity, and it's not just hard work everybody is putting in. Right? The maze through which they had to navigate, and this is tricky times, right? This is geopolitical complexity, then there is logistical complexity, then there is operational complexity. So this credit really goes to them. So I would like the organizing committee to sort of be, starting from you, madam, please come. Yeah. Because there's so many people are there, so you have to call them. I may not even know all the names. Come. This is your last chance to find out whether you really worked or not. Yeah. No. You cannot disagree with what I said. No.
I agree with Mohan, but more than me, it's the entire gang here. And as I said, it's Kritika who has done, that SGBC email which you were getting, it's this team, including making the brochures, the accommodation. Roshan has been very good. I think no issues with transportation. So I'm glad now we have a team who can do this. And they've also offered that, you can hire them for any event management. They are available.
That I do not approve.
But for me, thank you, everyone. This was not a work invitation. This was personal invitation, and I'm glad all of you made it. So thank you. We hope to see the next BRICS meeting soon. Where is Jose? Oh, there, Brazil. Thank you.
Thank you very much.
We'll go for lunch now, and the bus for Show Temple will be ready by 3 o'clock. So from 1 to 3, have your lunch. If you have not seen the center, see the center. And few people are part of a meeting that you will be guided to go there. And Paul is going for some fresh air. Okay, thank you.
Sorry, just a quick thing. I would like to thank the audience for being here. I would also like to thank the external participants for their wonderful posters and asking really good questions. So thank you for being a part of the III BRICS Symposium. Okay guys, so the bus will be there at 3 p.m. Have a nice lunch. See you all. Bye.